Uh, Good morning, everyone. Welcome, everybody, to the IBS conference of Nanomachine for Biotechnology and Medicine. And it is really fortunate for us to organize this meeting and invite all the prestigious speakers over the world. The director of IBS Center for Nanomedicine and a professor of Yonsei University and also the forum organizer of this conference will give a brief welcome remark.
Thank you, and welcome everyone for this quite amazing nanomachine conference. This is a series of IBS conferences that we organized over the years, and I'm very pleased to have all the distinguished speakers and audiences from the world. Let me briefly introduce the IBS. IBS is a institution that was founded about 10 years ago, and then in fact we are going to have a 10th anniversary coming up in next week, and it comes with a mathematics, physics, chemistry, and life science, and earth science, and interdisciplinary sciences, and then we promote the excellence in basic science, and we hope to be one of the top 10 world global leaders in basic science in 10 years. And we have about 30 centers around the country. And I'm one of the directors of the centers, and I'm located in Yonsei University. And Yonsei University is one of the prestigious private universities in Korea, and we started as a medical school in 1885, and now it became one of the largest universities in Korea. We have 14 colleges, 60 departments, and 38,000 students. It's a big university, and Yonsei University was happy to host IBS Center for Nanomedicine, and our research goal is converging nanoscience to interrogate biological systems with a precise manner, and we also want to be one of the world-class interdisciplinary research hubs of the world. And the theme of our IBS conference is about nanomachines, and it is for the medicine and also biotechnology. And probably you know a lot about famous sayings, and one of them is by the Richard Feynman. He mentioned about the nanomedicines for medicine, and it is simple. The quote is quite simple. What if it were possible to construct tiny machines designed to perform surgical operation inside the body? And then medicine would be revolutionized. So there has been many interesting discussions over the past 6 decades. And one of the breakthroughs that come into this area is molecular machines. And there are a number of supramolecular chemists who have developed a very interesting nanos or molecular structure that can do a wide variety of motions. You can do the shuttling, and you can do rotation, and other capability that needs for the mechanical components as a system. And but still, it's not there where we see cells and nanomachines around it. These are not yet achieved yet. So we can say that it is. The area is still in its infancy, and here are some of the examples that probably we are going to discuss today. And there are a number of wonderful concepts and methodologies to make the breakthroughs in this research area for the past a decade or so. And people have been developed the models, proportions, and some of those come with the navigation capabilities. and there are multimodal mode of actions, gripping and straining and stretching, and sometimes it comes with a group of the nano robotics and so on. But still, the field in general is not up to the level that we want to have. So we all believe that there will be more interesting system or components or devices with many hopes, but we'll see simultaneous challenges and also endeavors to make this possible. So with that, I'd like to introduce 8 distinguished speakers around the world, from Joseph Wang and Li Zhang and Shanhei Zhao and Brad Nelson, Samuel Sanchez and Perry Fisher, Dave Gracias and Hongsu Cho and me. So we'll have a somewhat small but very focused discussion on this topic today, and I hope you all enjoy this wonderful IBS conference. Thank you.
Okay, so I am Jaehyun Lee, and currently an assistant professor of Yonsei University and a research fellow of IBS CNM. And then I am chair of this morning session. And there should be 25 minutes of talk and 5 minutes of Q&A session. So I think we need to all be very punctual. So before 3 minutes to the end, I will give you a sign, a ring bell like this. So then I will let you know when you have to finish. So the first speaker of today is Professor Joseph Wang. Joseph Wang is a distinguished professor and the chair of the Department of Nanoengineering at the University of California, San Diego. And then he is also served as the director of the Center for Bioelectronics and Biosensors of Arizona State University before he joined UCSD. And he published numerous scientific papers and many books and patents. And then he received 2 American Chemical Society National Awards. He is also 5 honorary professors from Spain, Argentina, Czech Republic, Romania, China, and Slovenia. So today's talk, his title is Micromotors Go in Vivo, from test tubes to live animals. Please join me, welcome Professor Wang.
Thank you very much. It's actually good to be back in Seoul. This is from my visit 3 years ago. I was staying at the Plaza Hotel, so I love Seoul, as you can see here. It's a great city, good to be back even virtually. So I will talk about my journey, our journey, the whole group here, from early motor design to test tube bench, all the way now to in vivo animal studies that we will see during the day. So the early work was focusing on the propulsion, on designing a new motor, a new navigation. Now currently we have 6, 7 propulsion mechanisms, but starting with the magnetic work and ETR, and then the Penn State nanowires, and now we added the rocket, the ultrasound, the light, and the electrical propulsion. Lot of capability, and this led to growing sophistication, lot of contribution from around the globe, led to major new improvement in terms of power and speed, precise navigation, cargo loading, intelligence, collective behavior, multifunctionality, and biocompatibility. All of this new capability offer now diverse biomedical application and opportunity, from drug delivery to surgery. And again, 50 years after the fantastic voyage, we have realized this in vivo with a live animal using synthetic motor. I will talk about our work in the stomach in 2014. And a lot of this application was summarized recently in Science Robotic in my review with Lia Feng Zhang, talking about the use of different propulsion mechanism for variety of application. And a lot of the speaker will cover this topic. And we're all facing various challenges to realize this fantastic voyage, starting with operation, propulsion in the complex biological fluid, performing variety of demanding complex tasks, issue of biocompatibility, safety, access to various regions of the body like the brain, how to track the motor in real time with good resolution, and how to fabricate this in a scalability. And our group is focusing on oral drug delivery, which is the most convenient and preferred administration route for patient, although it has limitation of low bioavailability, poor absorption, and degradation of the drug. So we will try to address this limitation in this coming 20 minutes. Again, this is go orally through the GI, the stomach, or the various GI region. But we try to introduce the dynamic delivery compared to the passive delivery, and also to reach to different location. Again, the early work, this is 13 years ago, we play with cargo loading, navigation of the cargo in a microchannel level, in a chip network. So this was 2008. We can navigate the cargo, which is just a sphere in the narrow channel, showing nice navigation in microchannel using peroxide as a fuel. Then in 2008, our group and a group in Dresden of Oliver Schmidt, Samuel Sanchez, and Yongfeng Mei went to replace the nanowire into this micro rocket with this bubble propulsion that now can operate in biological fluid. The nanowire were limited to low ionic strength. They are not practical. Still using peroxide, we get oxygen bubble propulsion with a lot of force and ability to functionalize this rocket surface. So we introduced in 2010 this template deposition of the rocket using a polymeric outer layer and platinum inner layer, all by template electrode deposition. And this results in very fast propulsion. This is 400 body length. These are 5 micrometer rockets, so 2 millimeter per second. You can see remarkable speed, some magnetic navigation using this polyaniline platinum micro rocket. Even we improve it further with controlling the morphology of the platinum, over 1,000 body lengths per second. So we have a lot of power, capability, lot of strength. We move later on 2011 to operation in a biological fluid. We show moving in untreated blood or serum. This is slower, but still a nice propulsion in the biological fluid. All of this capability led us to think, how can we apply this to improve the delivery, oral delivery in the GI tract? Commonly, they rely on passive transit with very limited drug absorption, with a lot of this is being lost and flushed away. We would like to introduce the active delivery using rocket or genus particle to improve the retention on the mucosal layer and improve the tissue penetration from improving drug delivery and bioavailability. So one limitation was the peroxide fuel to operate in the GI tract. So in 2012, we introduced the zinc rocket that nicely operates using the gastric acid. So this is the same rocket shape, but replacing the platinum with a zinc layer. And also later on, we in a group from Wuhan introduced the magnesium one that operates with aqueous media in the lower GI. So the zinc rocket, again, here instead of oxygen propulsion, we use hydrogen propulsion using the proton of the gastric acid give you this simple high school chemistry to generate this hydrogen bubble. You see the zinc and the polyaniline or polypyrrole, and this allows us to operate in a acidic condition, picking cargo in very extreme low pH of 1.2, transporting and releasing the cargo. This was just to 2012. Later on, recently, we introduced this bi-component rocket where we have the zinc is in the back engine, and then we have the cargo in the front, and then we have a pH-responsive cap that this offers more specific and responsive cargo release. When the pH is changing, then it's open and expose the cargo, and the engine is pushing from the back. So all this capability led us to operate this zinc rocket in the stomach and genus particle in the lower GI. And this is summarized in this 2018 AFM review. So for the lower GI, we are using a genus, magnesium-based genus particle that also offer improved skin tissue penetration and retention. So in this case, you'll see later on the design of this micro rocket with PLGA with drug and chitosan. But early on, we focus on multifunctional zinc rocket that combine high loading capacity of the cargo with combinatorial delivery of different cargo. You can see different size of particle. And this is again autonomously released using this biconical design. It's break in the middle and the cargo is released and the motor is self-destroy. These are the distinct capability we would like to have: control the loading, self-destruction, biocompatibility, and autonomous release. Now with this offer now, going to the in vivo, we started this to play in 2013, 2014 with the stomach using the zinc rocket, and the GI 2015 using the magnesium-based motor. So again, in 2015, we used the zinc rocket and demo improved retention using gold particle to visualize. We show in live animal greatly improved retention on the mucosa layer using this proton hydrogen propulsion in the stomach. And simultaneously, we started to look at the lower GI, where we cover the motor with a protective coating in the stomach, and then it's dissolved. This is pH responsive coating to operate in the GI itself and to control the positioning different region of the GI to improve therapeutic efficiency. So the way we prepare this, as I mentioned, we use enteric coatings that protect the motor upon passage through the stomach. This is pH responsive. Once its pH increased to 6 or 5 to 6, the coating is dissolved and exposing the magnesium and start the propulsion, actuate the operation in the upper GI. And all of this capability led us to these 4 studies. Again, the improved retention. This is the first in vivo. Then we show that this operation in the acid also causes neutralization of the stomach, which is commonly used with a drug to PPI to neutralize. The motor offers dual functionality of both neutralizing the stomach, and then we have pH responsive drug release. Then it led to improved therapeutic efficiency against H. pylori bacteria in the mucosa layer, and also operation in the lower GI by precise positioning along the gut, upper GI, middle GI, lower GI. So we will discuss this next 5 minutes, where we in the stomach, again, we use the zinc motor with the conical shape, improve the penetration into the stomach wall and the retention of the antibiotic. as we mentioned, we use this also with the genus particles that can also operate in the stomach. As I mentioned, it can cause in situ neutralization of the stomach. So basically, we're depleting the proton, this reaction of the magnesium, and neutralize the stomach. And once the pH reaches 7, then the drug is released. The drug is in this pH responsive coating. So you neutralize the stomach and release the drug, all these dual function simultaneously. And this shows that we can optimize with the number of motor, we can optimize the time needed to neutralize the stomach. So 5 milligrams of this in the live animal. And here we show the improved retention of a dye, DID, compared to passive particle. These are polystyrene compared to magnesium. So greatly improved retention compared to passive with the same coating, with the same dye loading, but very poor retention compared to the active delivery. So all of this led to our first study to in vivo treatment of H. pylori bacteria infection in the stomach using antibiotic, claromycin. So we immobilized the claromycin in the PLGA particles. So we have again the magnesium layers, and we have ALT coated, and then we have the drug in a PLGA layer and outer layer of chitosan to improve the adhesion. So we nicely, these are 10 micrometer magnesium, and then we have this 3 layer of titanium dioxide to give you propulsion, drug loaded in the PLGA and the chitosan to improve the adhesion. And in this offer greatly improved therapeutic efficiency. Let's see if this video is working. Yes, so this is typical propulsion in the gastric acid. And again, from this Nature Cam paper, you see the improved therapeutic efficiency compared to the various controls. Subsequently, we went to operate in the lower GI. As I mentioned, we use pH-responsive enteric coating to protect and shield the motor while passing through the stomach. And once you enter the upper GI, the coating dissolve, it's pH responsive, and we can activate the position by controlling the thickness of the enteric coating. We can control the position of the actuation. So with very thin coating, we have very rapid actuation exposure. This is the medium coating, medium thickness is the middle GI, and the thicker one take about 2 hours until it dissolves its operation in the lower GI. So we can use it for precise actuation and retention in different sections of the GI tract. As you can see here, where we quantitated with the thin coating is in the upper GI, this is the medium one, and this is the thick one. As you see in this video, let's see if it operates. So it's passing the stomach, now the pH is changing, then you expose the magnesium particle. Let's see it again. You can pass shield it in the stomach, then expose it, and you see the bubble propulsion in the GI tract. So this capability led us to a variety of therapeutic applications from different drug delivery, nutrient delivery, more recently vaccine delivery, where we, in this case, we improve the immunity, we deliver alpha toxin, and then we generate IgA antibody. Again, the active delivery give you greatly improved production of the antibody for enhanced immunity. So the way we generate it, it's again magnesium with titanium dioxide to give you the genus structure. Then we put the toxin in a red blood cell membrane. It's collaboration with Liafeng Zhang. And then we adding the chitosan for improved adhesion, and the enteric coating for protection in the stomach. So it's passing through the stomach, then the terracoating dissolve and then exposing the magnesium for improved retention and delivery. So overall, you can see compared to static, these are again polystyrene, very poor retention. With the active motor, you see the retention along the GI. And these are the antibody production, where you have the red one is the motor compared to the static one. So greatly improve production of the antibody compared to the static. All of this capability also allow us to develop a transient motor that they disappear upon completion of their task. So in this case, the magnesium and the coverage, it's fully disappear within 15, 18 minutes. And this is all non-toxic, so nothing toxic is left. In this case, we have magnesium with zinc oxide layer. Instead of titanium dioxide, we have the zinc oxide. And you can see after 1 minute, the magnesium is already depleting and stopping the propulsion. In 1 minute, we still have the zinc oxide shell that disappear after 12, 15 minutes. So overall, it's all a fully degradable transient motor that fully disappear upon completion their task. As you can see, both the core and the shell disappear in these 15 minutes. You can see it in this slide where you have, instead of zinc oxide, we have silicon shell. So again, you see in 2 minutes, you see most of the magnesium disappear. And then after 2 hours, the silicon disappear and everything fully disappear. We can control the transient behavior. We control the time of disappearance and the lifetime of the motor. And so more recently, we are moving to pill because this is the normal way to deliver a drug using pill in the pharmaceutical industry. We work with the pharmacy, we make a pill matrix, and we can incorporate the motor loaded with the drug in a similar way that done in the pharmaceutical industry. You can see the propulsion generate as soon as the entering the stomach or the GI. You can see the propulsion of this genus particle. And the template, the metrics of this do not have any effect on the propulsion behavior. So we can mass produce thousands of these pills with the motor of different size, depending on the animal. If it's small animal like mouse or pigs, we work on a big animal model. And again, this shows the improved retention, in this case using a DID, compared to the use of the free motor, the same amount of motor. So we are adding the pill, we have 2% of the motor with the dye, with the cargo or the drug. Here is comparison to silica pill, where this is instead of active magnesium, now we have silica-based, the same loading of the payload of the dye, very little intensity. This is even better than the free motor, but this is very common delivery. This is the convenient delivery. So even compared to the free motor, the same amount, the pill offer improve retention of the payload on the stomach wall. And more recently, we're moving to large animal like pigs. But in this case, we separated the drug from the motor. So we call it microsterer pill, which in this case, we are we are not compromising the drug loading. We can use the same drug loading of 5 or 10%. We are only adding 1 or 2% of a microsterer. So the magnesium genus acting as a steerer to improve the hydrodynamic, they improve the pill dissolution and improve the steering in the stomach or the GI to improve the retention. This is especially important for Parkinson's disease. We are working with our colleague for levodopa delivery to improve the treatment, as well as for cardiac application in the case of aspirin. So you can see here the in vitro study in Petri dish. This is the static pill where you have a static particle with the same dye compared to the active one with the magnesium. So again, we have a dual effect of the improved pill dissolution, improved steering around in the GI and the stomach, and we are not compromising the drug loading because we are separating the two actions. So this result in greatly improve retention bioavailability of aspirin, especially in short time, which is very important for cardiac emergency. So in this case, we're using this again. You could see the pill used for the large animal model, the pig, and we take the blood sample every 5-10 minutes interval. Even if after a long time, we still have improved bioavailability compared to the static pill or compared to the free drug. But most important is the short time scale where really the cardiac emergency is important for fast delivery of aspirin to the blood. So we are really moving to large animal and we are moving to different organ that I will tell you next time. So overall, we have all this capability of the microrobot that we talk about, and we will hear much more today during the talk from all our colleagues for different part of the body, from the bladder to the eye to the brain. So we will see excellent talk from Samuel Sanchez, from Pierre Fisher and Brad Nelson, and from Li Zheng about different part of the body. That all this is a collaboration of the communities that led to this amazing progress over the past 2 decades, and collaboration with the clinician, as you do in Korea. We have excellent clinical team in UCSD, and all this detailed biocompatibility study, toxicity, biodistribution, and going to the regulation for practical FDA approval. So really, this was a fantastic journey over the past 15 years. I started 2005 to play with this motor on the bench, and we are now in large animals. Still, we have a long way to go, and we will see a lot of progress in the next 6 hours. So again, thanking my student and postdoc. Here is Jian King. Jian King is now professor in Yonsei. She's in the audience. She is now J.I. Good to see you. We have Wei Ga. This is a graduate 2016. He's now in Caltech. Jinjin Lee is in Michigan State. Fernando Soto contribute. Is in Stanford. Ziling Lu and Tailin. They are back in China, and a great collaboration with Lia Feng Zhang for all this animal study on a cell membrane. So fantastic collaboration for 15 years with Lia Feng. This is our library, and again, thank you very much, Kamasada, and I love Seoul. Thank you very much.
Thank you very much, Professor Wang, and we can take a few questions from audience. Okay, Professor Chan. Yeah.
Joseph, thank you so much for your participation, and I'm really happy to have your wonderful talk. And I have one question about your micromolar design. You have a active ingredient, zinc or magnesium in the core, and you have a Janus TiO2 or zinc oxide, and then you have a payload, and then a chitosan. So, how do you control the Janus capability? In other words, do you have any certain percentage of TiO2 coverage on the surface of the sphere, for example?
Yeah, we control all the factors, the sputtering of zinc oxide, titanium dioxide, the concentration, if it's the PLGA with the drug or chitosan, and the time of interaction. This is still need to improve the scalability. It's still one of the bottleneck is scalability of this for actual practical clinical application. But these are highly controllable and reproducible indeed. Thank you.
So do you synthesize this in liquid medium, or do you do in gas phase synthesis?
No, it's combination of sputtering in the clean room, and then we move to liquid for the different solution.
Oh,
yeah.
I see. Okay, great. Thank you.
Thank you.
Another questions? I think we can take one more question. Okay, Dr. Lungeli.
Thank you for your very nice talk. I have a general question. So these chemically propulsed nanomachines, do you see any other application than the GI tract? Could you imagine them, for instance, in the bloodstream or any other type of fluid of the body?
Well, this is a good question, and we picked this not because it's easy, but because the drug delivery is usually carried out in the GI. So the GI is really the open to the body. We all take drug orally, so we use it, but we are moving to different parts of the body, not necessarily with this one, although you can operate them in different sections. But the main advantage is stomach and GI, but we publish several, and we are moving to in vivo study in different part of the body, but and not publish and not discuss using different platform, not the magnesium or zinc. But we will see a lot of exciting talk in the next 5 hours of different in vivo applications from different motor, different groups. Yeah.
Thank you.
Okay. Thank you, Professor Wang. And then let me join in please the Professor Wang. Okay, so I will give you a few announcements so that you know from the YouTube participants. I think if you have any questions, you can type in a chat window, so I can read those kind of questions for the speakers. So yeah, you can type any kind of questions. And then also the second announcement is there will be a group photo session at the end of this morning session. So all the speakers, including the online speakers, please remain online until the morning session is over, so we can take a group photo all together. So thank you. Okay, so the next speaker is Professor Shanhe Zhao. So he's the professor of mechanical engineering at MIT. And then his mission is to advance science and technology on the interfaces between humans and machines for addressing grand societal challenges in health and sustainability. So he's the recipient of the NSF Career Award, ONR Young Investigator Award, SCES Young Investigator Medal, and many other awards. So today his talk is about merging human-machine intelligence with soft materials technology. So please join me, welcome Professor Shanhe Zhao.
Thank you so much for the introduction and the kind invitation. I really hope I can join this conference in person, but due to the COVID situation, we have to do this online. But I really look forward to further interactions with participants of this conference. So I'll talk about merging human-machine intelligence with soft materials technology. Chen Hezao from MIT. The opportunity we see is this gap between human and machines. Over the last century, we see great progress in terms of understanding and engineering human body. Modern medicine, biology, genetics, these are the examples. Similarly, in the domain of machines, electronics, computer, internet, AI, robotics, we see great progress here as well. However, there is a huge gap in between. And I believe, one theme of this conference is really about how to bridge this, great gap. The impact can be on better health, can be on understanding the brain, can be on, extend the human capability. So many great impacts on the academics and the society. This is a grand challenge, right? Just due to this dramatic different material properties between human body and machines. Most of the tissues and the organs in the human body are soft, white, living. On the other hand, most of the machines are really based on hard, dry, and inorganic materials, right? How to merge them together, right? This is a grand challenge in mechanics, materials, and biology. Now, if we take a closer look at the different components of the human body, except the teeth, bone, and nails, all other components are soft materials. Their rigidity is on the order of 1 kilopascal to 10 megapascal. They are also white, containing 70 to 90% water. They are also living. They can grow, sense, respond, self-heal. Basically, they are living hydrogels. These hydrogels are also very robust. Under millions of cycles of load, they still maintain integrity and well-being. Right? So then, based on this understanding of different components of the human body, we propose to use soft materials technology to merge human and machines. The idea is we design soft materials, especially hydrogels, with very similar mechanical and physiological properties as different components of the human body. And there are already fundamental studies showing that such hydrogels can maintain long-term high-efficacy interface with the human body. At the same time, we make this hydrogel interface extremely robust and adhesive so that you can integrate external machines such as computer chips, such as robots, such as sensors, actuators, into this hydrogel interface. They use that to form this long-term high-efficacy interfaces between human and machine. Of course, the study will base on decades of fundamental research on polymer chemistry and polymer physics. These are pioneers, some of the pioneers in the field. Right? But we also need to design extreme properties of soft materials. Basically, imagine this piece of jello, with similar amount of water as the human body, but we need to design this jello to be tough, to be resilient, to be fatigue resistant under millions of cycles of load, and also adhesive to both the human body and to external machines. Also active, because the human body is an active organism, so we need active soft material interface to interact with the human body. And today's talk, I will focus on the last property, active property, just in the spirit of this, nano machines. Even though I will most discuss micro and milliscale machines, but I think the spirit, along the theme of this conference. So in particular, I will discuss magnetic soft robots in the human body. Right, there are many motivations to use magnetic actuation to basically for drug delivery, for treatment of the human body. Professor Wang just gave a fantastic talk. Cover some of those reasons. Right, magnetic field can be remotely applied. The actuation is untethered. Magnetic field is safe to the body and shielded by the body. And also there are existing infrastructures to generate such a magnetic field. For example, Steel Texas is one company generating such a static magnetic field to control robots, to control objects in the body. We are now the first group, working in this field. There are many pioneers, Professor Nelson, who's in the audience, Professor CT, Professor Wang, who's in the audience, Professor Abbott, Professor Dupont, Professor Velastri, Professor Fisher, who is also in the audience. Already many pioneers already in the audience of this conference. I really look forward to their talks. But we made one contribution in this field. So in 2018, the idea is we proposed printing ferromagnetic domains for magnetic soft robots. Right? Previously, the magnetic soft robots, usually the magnetic domain are relatively simple. Right? Simple domain or a simple magnet or a few magnets. We propose this 3D printing method. Basically, you embed small ferromagnetic particles inside a soft material matrix. This matrix can be elastomer or hydrogel. Then you couple an electromagnet around the nozzle of your 3D printer. Then during the printing process, you can polarize, you can reorient those ferromagnetic particles so that you can program the ferromagnetic domains in this 1D fiber you printed, then in the 2D sheet and the 3D shapes that you program. You can program very complex ferromagnetic domains as well as the 3D shape. Right? Then another contribution we made to this field, I believe, is basically theory and model. Yeah. So here we develop a theory that can quantitatively model magnetic soft robots. Right? In this continuum body, you have lots of magnetic particles. How to model this accurately? Basically, we develop a model that can accurately predict the behavior of this magnetic soft robot under different types of magnetic field. Right. So here is a stress tensor we added to the Cauchy stress that can take into account of the body force and the body torque. You can see beautifully match between experimental and the theoretical results. I would emphasize the importance of such a continuum level model because the model can guide the design and later guide the control of such robots. And I will discuss both topics in later part of this talk. So with that, we can design and print different robot-like materials. Here is one. This is a simple 1D fiber, but we can program the ferromagnetic domains in this alternating pattern, and then we do simulation, right? No feeding parameters, just do model to do the design multiple times. Then once you determine this design, you print once, you achieve this this fiber under external field. This fiber deform into this interesting M shape, right? Then this is a 1D, 2D sheet. Similarly, you can do macro level of design using the computer. Once you determine the shape and the ferromagnetic domain patterns, you print once. You have this 2D origami. Basically, you can pop out a 3D structure from this 2D origami without using hand. So basically, magnetic field can control the deformation of these 2D sheets. Then 3D, so this is an organic structure, basically materials with negative Boson ratio. So we take advantage of rotation of this ferromagnetic domains within the magnetic field. You can see the rotation will shrink this material from both directions. That gives you this interesting organic behavior without applying compressive or tensile force on this material. So you don't need to contact this material. Just apply external field. You see this interesting negative Boson ratio behavior. Right? So then we can even think further. In our daily life, actually, we are programming data by programming ferromagnetic domains on this 2D hard drive. Right? Similarly, we can use a similar principle to infuse data model with 3D printing method into this 3D magnetic soft robot. Right? For example, here is one design. We can basically infuse this data and information into this 3D printed soft robot. And since we know how to do the model, we can use a model to validate this experimental results, or use the experimental results to validate the model to have a comprehensive understanding of this robot. Then we can deploy this robot in field. For example, here is one example. The robot can crawl. The size, as I mentioned, is millimeter scale. Later I'll discuss some, micro scale results. It can roll. This one, you apply a rotating field. So this robot can roll around. And then, it can stretch legs, roll back. And it can also carry payload. For example, here, as one example, we carry a drug pill. So, this robot wrap around this drug pill and then carry it to a desired location and to deploy the drug pill. So that's that. Nature called this a quick change artist, right? Because this magnetic soft robot can really change its shape within a fraction of 1 second. So before the pandemic, this is actually an exhibition of a London Science Museum. And London Science Museum, one visitor asked a very interesting question. Could a squeezy robot save lives? And our work has been focused on this task since the first paper. Right? So one particular problem or clinical indication we focus on is this ischemia stroke, right? A stroke is the #1 cause of a long-term disability, #4 cause of death in US. Every 40 seconds, someone has a stroke. Every 4 minutes, someone dies of stroke. So this is a really great, societal impact. However, stroke can be reversible if you can treat it within a golden hour, right? So, and after that golden hour, time lost equal to brain loss. How to treat a stroke? Here is one, conventional method, which is called, guide wire. So this guide wire, you can see the shape is a J shape, and then the doctor operates the guide wire under a fluoroscope. They will make an incision on the groin of the patient, and they insert this guide wire from the leg all the way to the brain, and they use microcatheters, other stents to do the treatment. So that's the idea. Then you may ask, how do I navigate the complex branches of blood vessel? Right? Because the shape, the tip of this guide wire is J shape, so you can rotate the guide wire from outside, and the J shape will rotate inside. Then you can navigate this guide wire. Lots of challenges. #1. Loss of maneuverability under large friction. You can see this is a very long pass from the leg to the brain, right? Of course, there are friction accumulated, so you can lose maneuverability due to this twisting motion. Unavailability of doctors. This is intrinsic challenge. It's just impossible to put a very expensive neurosurgeon to every small hospital in small towns, right? So they are all usually in those tertiary, large medical centers. Then the third one, for these heroic doctors, they suffer a lot. They suffer from accumulated radiation from X-ray fluoroscopy, right? For the patient, it's one treatment, right? After that, the patient is gone. But for the doctors, they may need, multiple operations per day, right? Throughout their career. So they worry a lot about this accumulated radiation to the doctors, right? So we propose a strategy. The idea is, highly operated, even autonomous robot that can navigate within the blood vessels of the body and the brain. The idea is now we print or we fabricate this guide wire robot with this programmed ferromagnetic domain. And then once you apply external actuating magnetic field, this guide wire can bend on demand. Then you form this J shape on demand towards the desired branch, desired navigation direction. Then with that, you can really navigate on demand by controlling external magnetic field without rotating this guide wire from outside the body. Right? We further develop a hydrogel skin coating on this guide wire. The hydrogel skin coating can reduce the modulus of the surface of the guide wire by 100 times, friction over 10 times. So basically, further reduce the friction. Even though we do not need this rotational operation, we further reduce the friction. Right? Then with that, we further, program or design this magnetic polarity patterns on the tip of this guide wire, so that the guide wire can reach the largest workspace, basically to achieve the most agile guide wire within the blood vessel, right? Because we can program the magnetic particle concentration in each voxel of this magnetic guide wire. So we use that as our design parameter. Then the desired function or the functionality we want to achieve is this largest workspace, the largest area the guide wire can achieve. Then we use this genetic algorithm to do the design. Basically, we randomly generate many possibilities of the polarity. We calculate the workspace. Now you see the importance of this model, right? We cannot do experiments here. We basically, based on the model, calculate the workspace, and then we use a stochastic universal sampling. Then use elitism, crossover, mutation. These are standard algorithms in genetic algorithm. Then you iterate this multiple cycles. Eventually, we come up with this very unintuitive patterns. So you can see here you have around 25% magnetic particle concentration, here 40%. Then in between you have 0%. On the very tip, another 40%. Right? Very challenging to imagine, such a design. But with the genetic algorithm based on our model, we can achieve this optimized polarity pattern of this magnetic guide wire. Then with that, we can deploy this first in phantom models. Here is the result. So here is a real size cerebrovascular phantom with simulated blood. The phantom is made of very soft silicon, and inside we infuse this pulsar type blood flow, simulated blood, so to really simulate the realistic environment. So you can see these are 3 aneurysms. One point I want to emphasize, this catheter can really avoid touching those aneurysms. So that's the beauty of this magnetic guide wire. So here are the 3 locations of this aneurysm. You can see. Then in comparison with a passive guide wire, right? So here is what a neurosurgeon would do, use the passive guide wire. Due to this J-shaped tip, passive guide wire unavoidably will touch blood vessel, will touch the aneurysm. So we collaborated with many neurosurgeons and neurointerventionalists. That's the feedback from them. And here you can see, because this sharp turn, the radius of curvature is really small, so this guide wire wouldn't be able to navigate across this. So then the neurosurgeon have to pull out the guide wire, reshape the tip, insert it again. On the other hand, for this magnetic soft robot, soft guide wire, right, it can on demand bend the guide wire. And this radius curvature is controlled by the external magnetic field. So you can really smoothly navigate within the blood vessel. So that's that. Then let's see some realistic system. So we further develop this integrated system. We really think this is the most challenging part really of this project. We integrate a C-arm fluoroscope, one robotic arm controlling an external magnet, and this magnetic guide wire, which is improved version. We use polyurethane to make this really medical grade quality of the guide wire, very robust and very stretchable. Then with that, we deploy this into a phantom model first. You can see the patient is here, and then we navigate this guide wire. So here's the phantom model. We use a joystick to control the advancing and the retraction of the guide wire and other microcatheters. So the whole system are robotically integrated. And here you can see this basically within this virtual environment, right? You can even work this robotic arm with biplanary fluoroscopy, right? Very accurate medical images. And this is a real-time navigation of this guide wire within this complex vasculature of the phantom model. So that's that. Then we try to, to navigate this under fluoroscopy navigation. So this is the image from the fluoroscope. You can see this guide wire is can be easily visible under this fluoroscopy, and then you can navigate the external magnet, right, to control the navigation of this guide wire within the blood vessel. Then we actually test some clinical indication. One important task is this embolization coiling, this aneurysm embolization coiling. So here is the aneurysm. We navigate the guide wire to this aneurysm, and then we deploy a microcatheter, all with this robotic system. Then we can even do this coiling with the robot. So the doctor actually doing all this operation in another room. The doctor does not need to operate beside the patient. This is the second one. Here is a clot within this blood vessel phantom. And then we use this guide wire under the control of an external magnet. Pierce through this clot first. And then we deploy the microcatheter here through the joystick I mentioned, the advancer retriever, to deploy this microcatheter. And then after that, we withdraw this guide wire. Oh, sorry. Then we deploy a stent retriever. So the stent retriever is something neurosurgeons use to retrieve those blood clot. Then together with the microcatheter, you retrieve this blood clot. All this robot, the neurosurgeon is operating this in another room, right? So that's that. Then we further move another step further. This is in the pig model. Sorry. And we use a very mature protocol of this pig elbow, musculature anatomy to approximate the complex musculature anatomy within human brain. Right? So you can see this 3D angiography of the 3D architecture of this blood vessel, very complex 3D architecture. Then this is the real-time deployment of the guide wire. So to our knowledge, this is the first time of such a soft continuum guide wire without magnets, with dispersed magnetic particles navigating inside a living animal model. And this beating sound is the heart beating of this pig. You can see, successful navigation. And also, this is a very intuitive operation. So you can see the learning curve. So this neurosurgeon really loved this. So we work with a team of 6 neurosurgeons. You can see, the 2 students are very experienced user of this system. So they can do this task within a very short time, 50 seconds. The neurosurgeons, you can see, very fast learning curve. After 10 trials, they already reach experienced user level. So to them, this is amazing, because for manual operation, they cannot achieve many of those tasks, and it takes a very long learning curve for them to be experienced. With this system, just over 10 times, they become very experienced, very comfortable to use such system. And so now we are further moving ahead to make semi-autonomous, even autonomous navigation of such guide wire and microcatheters within the blood vessel. I do not have time to discuss details about this, but I do want to discuss this vision. AI plus 5G plus robotics. Many people think this will give us an autonomous car, but there can be very much societal issues, right? How about so many Uber and Lyft drivers? How about pedestrian accidents, right? There can be lots of societal issues. On the other hand, we believe AI plus 5G plus robotics will give us future medicine, especially in this highly operated autonomous medical robot, right? Because scientists, we are on board. Neurosurgeons, they are actually totally on board, because we are not replacing neurosurgeons. We are enabling them. They can save more lives remotely. Patients, time loss equal to brain loss. You can see all the stakeholders are on board, and the technology are converging towards this vision. So with that, I will conclude. I discussed 3D printed magnetic soft robots. Extremely important is this quantitative model for this continually deformable robot. And I discussed the vision of highly operated, even autonomous robot within the body. AI plus 5-6G plus robotics. We think this will represent the future of medicine. If you want to know more information, you can take a look at our group website. Then acknowledgement. So I want to emphasize this student, Kim, really leading the whole project. He has lots of frustration, to fight with reviewers of his papers, but he's just a brilliant, talented student. And the others, Rick Zhao, currently assistant professor at Stanford University, previously working on the modeling part of this. Collaborators, I want to emphasize Professor Aman Patel, who is the chief neurointerventionalist at Harvard Medical School. So he agreed with our vision, collaborated with us thoroughly throughout the project. And I want to thank the founders and thank everyone in the audience. Thank you very much.
Thank you very much for the wonderful talk, Professor Zhao. And then we can take a few questions from the audience. So I can ask some simple questions about your first part, magnetic robotics. Do you think for now is the size is a little in a centimeter scale? So can we actually do the same performance in a nano or micro scale robotics?
So for magnetic robots, right, the general field of magnetic robots, the answer is yes. I believe Professor Bradley Nelson in this afternoon probably will discuss some micro and even nanoscale robots. Just beautiful work, to achieve potential functions. The answer is yes.
Okay, thank you. And yeah, so thank you very much, Professor Zhao, and please give him a big hand for the nice talk. Thank you. So we can have like 20 minutes of break time. So right now it's 9:50, and then we can start from 10:10. So yeah, we have 20 minutes, and then let's get together outside.
Hello? Hello, can you hear me? Hey, Joe. Yeah, hi. You need to unmute. Yeah, but I can see your.
Okay. So I think you have a coffee and nice breaks, and then so now we have three speakers, and then first speaker is the Professor Li Zhang, and he is a professor in the Department of Mechanical and Automation Engineering, and a professor by courtesy in the Department of Surgery at the Chinese University of Hong Kong. So, and he's also as a program leader for the Multi-Scale Medical Robotics Center at the Hong Kong Science Park. And his research interests include micro nanorobotics for translational biomedicine and their collective behavior for development of intelligent small-scale robot swarms. So he have received numerous awards, including the Hong Kong RGC Early Career Award and several another award from IEEE conferences. So today he will give a talk titled with Magnetic Micro Swarm for endoluminal procedures. Please join me welcome Professor Li Zhang.
Okay, thank you. Thanks a lot for the nice introduction from the chairman. And I would like to also thank Professor Chen for the kind invitation. I feel very sorry that I cannot come on site for joining this conference due to the COVID. And you know, the problem is not that I come to Korea, is when I go back to Hong Kong, then you know, they are very restrict quarantine policy. Okay, the title of my presentation is Magnetic Microswarm for Endoluminal Procedure. So I'm going to show some results from my lab, how we generate and actuate the microswarm and use them for the endoluminal procedures. Next slide. Okay, so I think the first speaker, Joe, had given a very nice introduction, so I will not repeat here. So basically, over half century ago, then people started to realize that, you know, if we can make things small, then it makes sense for the medical treatment, such as diagnosis and also the intervention. Okay, I think one good example is actually from the development of endoscopy and how people realize that we can use some tools for the diagnosis of our GI system. So in this slide, you can see that, you know, in the 19th century or before the 19th century, basically people did not know how to make the device small to let them reach the GI directly. So here you see that people use some indirect way for the diagnosis of, you know, the stomach and the esophagus and so on and so forth. For example, you can use a barium meal, you swallow the barium meal, and then you use the X-ray, you know, for this kind of inspection. You inspect the shape of your stomach and your GI to see that whether there's some disorder or not. You see that later in the middle of, you know, the 19th century, then people first developed this kind of rigid endoscope. As you can see the image here, you know, because the endoscope is rigid, you know, basically the patient will feel, you know, huge discomfort during this kind of operation. And also from this image, you can see that, you know, because the light can only illuminate part of your stomach. So basically, you know, this tool has a lot of, you know, this trouble, right? And about 100 years later, then people developed this so-called flexible, you know, this fiber scope. So basically, based on the development of nanotechnology, optical fiber, and other, you know, this micro nanotechnology, then people were able to integrate this kind of soft, you know, these tools, you know, into the endoscope. And we also know that nowadays, you know, people can inspect the entire, you know, small bowel by using this kind of enteroscope, right? By using the single balloon or double balloon enteroscopy system. But however, it still takes a lot of time, you know, to complete this kind of inspection. And again, you know, the patient won't feel very comfortable because, you know, basically you deform the shape of the small bowel. So it generates, again, a lot of discomfort. And later you see that, you know, since the early, you know, 21st century, then, you know, an Israel company called Giving Imaging then developed this kind of wireless capsule endoscope. So basically, you know, the patients swallow this miniaturized capsule, and then, you know, this kind of tool can be used to inspect the entire, you know, this digestive system in a non-invasive way. So here you see that the development of endoscopy is really changed from rigid to flexible, and, you know, one tendency is to the wireless, you know, this kind of device based on the, you know, development of miniaturization. And about 3 years ago, you know, there was a very interesting editorial article published in Science Robotics, which showed that the medical robotics actually across all the scales, from a very, very large scale, that's the human scale. We know, you know, Da Vinci system as an example, a very successful example. And then we know that at the meso scale, then we also have this kind of endoscopic, or, you know, this catheter tools. Our last speaker, Professor Xuanhe Zhao, also showed his recent development of this kind of tools, which was very impressive. And if you further go to smaller scale, then you can see that many of these kind of tools are changed from tethered to untethered design. I think, you know, basically the main advantage of this kind of miniature robots is that they have some possibility or easy to access those hard to reach region for minimally invasive medicine. So here, what you see is actually a assembly image of capsule. So you see that basically the design of the capsule is still based on, you know, our microelectronics and also the traditional mechanical assembly approach. But we know that the same approach cannot valid, you know, if you make your device 1,000 times smaller, right? As we show here, you know, one of the kind of micro scale robot. Because you know that you cannot apply the same assembly, you know, strategy, and you also have the issue, for example, to find a battery. So that's the reason we're thinking that if we want to further miniaturize our device to reach the micro and nano scale, then we need to think about other ways. For example, you should use much simpler structures and more rely on the functional materials and the so-called physical intelligence, right? So this is one way. So alternatively, we can also think about to use many of this kind of simple agent and let them work together for performing, you know, complex robotic tasks. And this we call micro swarm. So that means we hope that, you know, by using the simple, you know, the structures, many of them, and they can generate higher level of function or even the intelligence for performing medical tasks. And also, you know, that in that editorial article published in Science Robotics, it shows, you know, 10 grand challenge in science robotics for the next 10 years. I think one great challenge is the design and development of robot swarm. So here you see that I show two papers, one published in Science and one published in Nature. You know, they are kilobots and particle robots at, you know, a centimeter scale. So our research interest is that how we can further, you know, make this kind of a device smaller to reach the micro nanoscale. So how to create, you know, this kind of swarm to understand their collective behavior and whether we can use them for medical application. Of course, there are a lot of pioneering work. So basically, we can still summarize them, you know, this kind of swarm to two types. They are, you know, decentralized system and centralized system. So one intuitive idea is to employ, you know, the nature microorganism and, you know, do some kind of control or maybe use their nature taxes, you know, for the targeted delivery purpose. For example, this is a result from Professor Samuel Sanchez's group. And this is a result from Professor Metin Sittis' group. So there's some result, you know, what I show here is from Professor Sylvain Mattel from Canada. So basically, so here what you see that you can use different type of microorganisms, bacteria, you know, use them their taxes. And sometimes you can also, you know, get help from the external field, such as magnetic field, you know, for the targeted, you know, this cargo delivery purpose. Okay, and of course, you know, another method is we can design our synthetic nanomotor or nanorobots for the same, you know, this purpose, you know, to generate a swarm. And of course, you know, it also can be classified to types by using either by use external fields such as magnetic fields, Here you see some kind of pioneering work from Professor Nelson's lab and from Professor Pierre Fisher's lab. So they both show that, they were able to control a swarm thousands of the helical micro nano robots in vivo, for the targeted delivery purpose. And, Joe also had published an interesting paper 6 years ago by using the ultrasound, a swarm can be generated. And, but these are the kind of centralized system, right? So regarding the decentralized system, then, the chemical field is a very popular approach, as, we show here. But due to a time constraint, I cannot show the details. So we're thinking that, the main advantage of using a micro swarm, or sometimes we call micro robotic swarm, instead of, control a single agent, is that, you can significantly increase the dose, for the drug delivery purpose, if it is required. And the second way is we're thinking that, for the imaging purpose, then, you get some advantage of that. You can do the real-time tracking, or, sometimes the imaging of, of a swarm as an entity. So that means, the resolution of the imaging modality is not necessary to be very high. But think about that individual agents, they're at the micro nano scale. Actually, nowadays, if you look at the clinical imaging modality, they can hardly reach that resolution. So the third advantage we're thinking is adaptability and also the robustness to complete the job. Because think about that, if you are using a single untethered robots in the dynamic environment, it can happen that, if one robot is failed, then basically the whole, the task will be failure. However, if you have 1,000, if 10 of them or tens of them are failure, then, you can still complete the job, right, without problem. So basically, we're thinking that whether we can generate and control basically thousands or even millions of these kind of nano robots, to deliver them in the lumen, and different part of lumen, for example, the blood vessel. And we are also interested in those kind of tiny lumen in the GI tract, such as a bioduct. So here you see, the shape of the bioduct is really look like the root of a plant, right? It has many branches, they are very small. Basically, the diameter is about 1 to 2 millimeter. So these are the kind of the interests, from our research lab. So we start from the thinking of, the building agent. So basically, the building agent need to be simple, small, safe, and functional. So that's the reason we use iron oxide nanoparticle, because we know that iron is a required element for our body, and that there's a very mature technology to fabricate iron oxide, right? Nanoparticles, we can tune their diameter. They are relatively safe. For example, they are used as a contrast agent for MRI. And, we're interested in controlling them by using a magnetic field, because we know that on one hand, human body is kind of transparent to the magnetic field. On the other hand, it's kind of safe to our body. So we first, prepare this kind of very simple 3-axis Helmholtz coil for our actuation purpose. We also use a very simple, this rotating magnetic field. So here, what you see that you put many particles in a small reservoir, and when you're applying a magnetic field, then the adjacent nanoparticles will create, will attract each other and form the magnetic nanoparticle chain. And when you rotate, the magnetic field, then basically the chain will start to rotate. And here sidewise, we put the ultrasound for the tracking purpose. So that here is the result show that, when we tune the input parameters, for example, by tune the field strength and also the rotating frequency, you can see some kind of phase diagram. So in certain range, then basically those particles will gather together to form some kind of vortex-like microswarm. So the main advantage of this kind of vortex microswarm that you're really able to gather majority of these particles to a very small region and control. That means navigate them as an entity to go through a kind of tortuous channel, as we show here. So if you change this rotating field, in the horizontal plane to the vertical plane, as we show here on the right of this video, you will see that, basically these particles chain will tumble and move individually. And when this kind of tumbling nanoparticle chain goes through the complex channel, you lose many of them when it reaches the target, right? So that's the reason we think, this kind of microswarm makes sense. And basically, the generation of this kind of microswarm is to understand, interaction force between the magnetic dipole-dipole interaction and also, the fluidics generated by this kind of rotating magnetic chain. But you know that one issue for this kind of vortex-like swarm is it actually can influence by the solid boundary significantly, because you need the, the liquid flow to generate this kind of swarm. So it's very understandable that, if you have a sidewall, then actually the fluidic force may not strong enough in order to induce this kind of swarm, right? So that's the reason later we change our field, from the rotating field to some kind of oscillating field, as we show here. You have the B vector, then basically you swap the B vector from left to right, as we show here. And then again, we see some kind of interesting emergent behavior. So here again, the horizontal axis shows the oscillating frequency of the input field, and the y-axis is defined as gamma. So gamma is basically the field is a ratio of this field is a field this strength. So here the ratio we show here is of AB and OP. So this is defined as gamma we show here. And we found that, in certain region, in this zone 2, for instance, then we can gather, millions of the particles together to form some kind of ribbon-like swarm. And interestingly, if you tune this gamma value, you can also generate some deformation. And it behaves like a rubber band, but please remember, they are formed by millions of nanoparticles. And we can do the navigation, so we can separate them to subswarm and let them go through the channel, and then, to let them gather together again. So this is we can realize in the DI water. And later, we also found some other interesting behavior to further improve, by further improving, the magnetic dipole-dipole interaction. We found that this kind of swarm can even create some kind of solid-like structures. Think about, this is a top view image. This swarm, basically, you look from the top view, it looked like, a planar, right? A very thin and planar plate. But now we are even able to, tilt it for 90 degrees. So here you see the change, right? I mean, you see the change. You see from the top view, it's flat, and you tilt 90 degrees. Then something like you can see it from the sidewise. It's very much like, you have a credit card. You look at from the front side, and then you look at from the sidewise, from the side. Okay, so we can even realize that by further improving, this magnetic interaction between the, this nanoparticle chain. And later, then we also investigate our swarm in the biofluids, including, the blood. So here the results show that, we're able to generate our swarm in the 4 times diluted blood. You see that the swarm can be generated. And here you can also see that, by using the ultrasound B mode, so we're able to track our swarm by using the ultrasound. So this is in the whole blood. So this is in the 4 times diluted blood. So you can see that the swarm can well generated. Basically, if you can tune the magnetic dipole-dipole interaction, improve the interaction force, then you can do something like that. But the one problem for the ultrasound B mode is, you can hardly get information from the environment. it's very noisy. So then later we are thinking that whether we can use some other imaging modality. So fortunately, by for the ultrasound imaging, there's another interesting imaging mode based on this kind of phase change contrast. So-called Doppler imaging. So then, basically what we do that, so we can use the Doppler signal to image, either the swarm directly, or we can also use the Doppler image to image, for example, the red blood cell, so which is moving, surrounding our swarm, as we show here. Yeah, because this kind of when this kind of red blood cells rotates, you can see that one side the particles, this red blood cells will move towards ultrasound probe, and the other side will actually move away from the probe. So that means, it can generate both positive frequency shift and a negative frequency shift, allowing us to create some interesting Doppler signal as we show here. So you see that one side is red and the other side is blue, implying that, our swarm is in between. Okay, is in between of. So this is a kind of indirect imaging, mode. But the main advantage is that, in this mode, then your ultrasound signal is not necessary, interact with our swarm directly. So basically, if your ultrasound probe has a little bit shift away from our swarm, we can still generate a signal, okay, because of the red blood cells. So that's the main advantage. And that we also found that, we are able to do the magnetic actuation, even with the, this flow condition. Okay, so here you see the result, by some kind of blood flow and also the pulsatile flow. Then you can see some kind of Doppler image signal, close to the sidewall. These are our micro swarm created by magnetic nanoparticles. Okay. So what's a goal, for this kind of research? We're interested in using for the, the stroke treatment. I guess I don't need to repeat the importance of the, this kind of treatment. Xuanhe had given a nice introduction. So the region we're interested is really in those kind of tiny and tortuous parts. So, the difference, I think, or the beauty of this kind of nanoparticles is we're thinking that maybe we can do the treatment not just at one site. So if multiple sites has a blood clot, then whether we can use our swarm or the nanoparticles, by using the magnetic field to deliver or deploy them to the, to the different regions simultaneously for the blood clot dissolving, I think that will be very interesting. Or, another way, so if you really go to, this kind of distal, which cannot be reached by the guide wire, then maybe swarm, magnetic nanoswarm can be an alternative choice. So currently we're using, we're doing a study of the, using the fluoroscope, for the real-time tracking of the nanoparticles. We got some result, but this is still under investigation. Okay. The next part in the last, couple of minutes, I want to show that we're not just interested in the, these blood vessels, the cerebral, blood vessels. We're also interested in delivering, the micro swarms to the GI, to the bile duct, for instance. So here we show the diameter of the lumens in this region, in the liver, bile duct and pancreas. You see that they all have very small diameter. And then, here we change our design from the nanoparticles to the cells. Because one advantage of the cell that, it can create, the miniature immune response. So basically, they can become biocompatible and biodegradable, assuming that the cells are from the host, right, patient. And, the cells can be soft. So we use a 3D culture approach to co-culture, here you see that the several 100,000 cells together with the magnetic particles. But again, the majority are the cells, as you can see here, 28%. And they are very soft, as soft as a brain tissue. And you see that this kind of, this cells fluid can go through, this kind of tiny channel, even with a very large deformation, so without problem. And another interesting thing that, this kind of cells fluid, can rotate on a surface. It moves like a solid when you apply a rotating field. And then if you apply some kind of magnetic field, to do the anchoring, then they can spread like fluid. So that's really a reason we think that, maybe we can also call them like a kind of swarm. There's a small aggregation like a swarm. This kind of spreading is basically a decentralized, right? You cannot control, fully controlled by the magnetic field, but this is basically is a behavior from the cells. And we found some interesting paper published in Nature Material before. The ants can also form this kind of sphere. And, they change their behavior from solid state to liquid state. And here you can see that the cells fluid after anchoring, then they spread, like how, a liquid droplet, wet a surface. And here we show that, by using the assistance by using the endoscope, we're able to, deploy our, this cells fluid close to the bioduct in the doodle. Then after deployment, we can change our imaging mode from endoscopic view to the ultrasound mode, to further, track our cells fluid in the bioduct. And so, the beauty of this work is we really show that we're able to deliver our cells fluid deep inside the body in the meter range in minutes with high precision. So precision can be, in the micrometer scale. Okay, and so this is really the summary of my presentation. We show that we are able to create, this nanoparticle swarm, and also the swarm by using the cells with the nanoparticles, and and use them for the endoluminal intervention with the help from the catheter and from the endoscope. So we are still working on that research project, and I would like to thank my group member and also those alumni who graduated from my lab, and also my collaborators from the Prince of Wales Hospital in Hong Kong. That's all of my presentation. Thank you very much.
Thank you very much for the wonderful talk, Professor Zhang. And then we can take a few questions from audience. Okay, yeah, Professor Kwok.
Oh, hi. Thank you for a nice talk. This is Min Seok from Yonsei University. I just have a question about your magnetic microswarm. So it's very impressive technology, but I'm just wondering if you can precisely control the size and number of those microswarm within the one system. I see that you can control you can make a big aggregates of a magnetic nanoparticle, but can you make like a double or triplet of them in the one system?
Okay, thank you for your question. So my understanding is that your question is that whether we can control 2 or 3 of them simultaneously, or?
Yes, that's correct.
Yeah, okay, so yeah, that's a good question. That I think this is one challenge for this kind of centralized control. Somehow, you have some chance, by tuning, this input frequency, because, many groups had shown that, for individual control of, several, individual microrobots, by using the so-called step-out frequency. So for the swarm, one way you can do this control is you actually can design different type of building blocks. For example, if one microswarm is created by magnetic nanoparticles, then maybe the second swarm can be created by magnetic nanorod. Or another way is that you can design your building blocks of nanoparticles with different diameter. Then that means when you're applying the same magnetic field, then you can naturally or automatically change their interaction force. Okay, so you have some chance to control, some of them, but it's difficult. Yeah. On the other hand, I don't think it's really, the advantage of this magnetic control. Yeah,
Thank you so much for your answer.
Thank you.
And we have a question from online audience, and then they are so one person is asking that your micro robot is composed of the carbonyl ion and not the Fe3O4, and then he is asking why. I think that it may be related to the magnetization value, something like that.
Iron oxide nanoparticle?
So the question is that your stem cell soft micro robots are composed of carbonyl ion, not the Fe3O4.
Oh, okay.
Yes.
Yeah. So actually, one of my ex-PhD student prepare those particles. So perhaps it's not that critical, because you can see that this kind of this carbonate, these ion particles are encapsulated in the cell, and the percentage is very low. And what I remember that, those particles, the diameter is also large, not in the real nanoscale. So I think, the main selling point is not the size or the material of this kind of particle. Is really we show that just by put little bit of this, this ferromagnetic material, then we are able to do the nice magnetic navigation of cells, several hundred, for example, the 200,000 stem cells, together, and, you're able to move the cells. Yeah, so this is what we want to show. But certainly, I think we can still change the magnetic nanoparticle to iron oxide. But then the force may be smaller. Yeah, yeah.
And then, what are the surface state of the nanoparticle to avoid some clumping and irreversible aggregation, things like that?
Yes, that's also a good question. I think, for those nanoparticles, naturally, they would prefer to aggregate. And the thing that, when you're applying the, this dynamic magnetic field, localizedly it creates some flow. And, so that's the reason we need a dynamic field, right? To keep them moving, and otherwise they will create aggregation. And another way is, of course, is to tune, their surface properties, create some kind of, the charge to let them, repel each other. But of course, in the biofluids, then it's not as good as that in water, or, because there's a high ionic strength. So it actually influenced by a lot of parameters. Yes. So we need to tune these things depending on the environment where, where you want to apply. Yeah.
Yes. Yeah. Professor Chan.
Thank you so much. I really enjoyed that. I have one related question. So do you think you can able to control the number of the cells in terms of the aggregations? Let's say you have a single cell to tens of hundreds of them, depending on the magnetic components of the nanoparticles, or the number of nanoparticles that you are infused into the cell. Is that correct?
Yeah, I think it's possible, because these cells, basically what we do, they're just a 3D culture. So it's really depending on, the culture condition, the time. Okay. I think we're able to control the cell number, from very small amount to large amount. Yeah. But the problem is that when you want a very large, the cells spheroid, then the cells inside of this spheroid cannot get sufficient nutrient, so they will die. Yeah.
Oh, I see. And then can you also try other type of cells instead of stem cells, red blood cells, and other immune cells, T cells, and so on? Is it possible? Have you tried?
We haven't, honestly speaking, we haven't tried it yet. But that basically, so these are kind of mature technology, the 3D cell culture. So we can just check in the literature what kind of cells can create, this kind of 3D spheroid. Or maybe even organoid, organoid, for the delivery. I think that's possible. Yeah.
Yeah. Right. Okay. That's great. Thank you.
Thank you.
Thank you very much. And then please give him a big hand for the nice and wonderful talk.
Thank you. Thank you very much.
So the next speaker is Professor Jinu Chan, and then you know that he's the organizer of this forum, and he's also the director of the IBS, Institute for Basic Science and Center for Nanomedicine, and he's also Horace G. Underwood Professor of Chemistry at Yonsei University, and his research focus is on the nanoscale phenomena of nanomaterials and their applications for biomedical fields. He also got numerous awards, including the University of Illinois Schiss International Alumni Award for Exceptional Achievement and Hoam Prize, and many others. And he also selected as Korea's 100 most influential persons for the next 10 years by Donga Daily, and world's most influential scientific minds by Reuters. So his talk today is Magnetogenetic Nano Toolkit for Molecular Interfacing for Neurosciences. And please, please join me. Welcome, Professor Chan. Thank you.
Thank you, and it is my great pleasure to present some of my work, and I really enjoyed the previous talks, and then it was phenomenal. And then my center has some, a couple of interesting avenues to explore. One is the nano imaging and communication. So we are developing molecular and nano contrast agent to visualize, the biological phenomena from in vitro to in vivo system. And then the second topic that we're interested in is the nanomachines, and we have learned a lot about interesting studies, in particular the remote mechanical nano actuators. That's something that we have an interest. But we are starting from the bottom up approach. So in other words, we design materials and we integrate them with molecular systems. So what we are interested in is we have cells with cellular receptors, genes, and so on, and then we have nanomaterials, and perhaps we have nano devices, and so on. We are interested in this part. So it's truly nano or molecular scale interaction that we are interested in. So we have been, my research group, because I'm chemist by training, we have developed a number of different type of nanoparticles over the past 25 years. So it is interesting, and it has a number of variety of chemical and physical phenomena. And then in these days, we are tuning into different avenue of new materials. We say it's designer nanomaterials. So it's designed for metastable nanomaterials, and that's in many part purpose-driven scale nano studies. So we call it functionally redesign. That means that let's say we have a nanomaterials, we can do tuning in terms of size, shape, composition, structure, and so on. But we are interested in some new phenomena associated with different structure and let's say porosity or the distance between the nanoparticles and so on. So we were able to find some interesting materials property they can readily utilize for biomedical studies. So today I'll tell you a little bit about how we can study the or learn some science from the nature. So we are trying to have some interesting model system from migratory animal, and then if so, we can get some some interesting understanding from the nature. Then can we really design a new tools to study biological systems with better precision? So here we learned a lot about magnetism, because magnetism is simply quite advantageous, because light is phenomena is amazing medium that we can utilize, but when it comes to the biological system, the penetration depth or interference with the tissues and cells are so severe that we cannot really go into deep. So in other words, one of the ways ultrasound or magnetism is also have been well studied, and we learned a lot today. And then here are the some interesting optical system that can explore biological system. We all know about the FRET. You can do a single molecular study, and as Storm said, you can really go into fine features of organelle and cellular level imaging. And the optogenetics, we utilize light to investigate neuronal behaviors. So what we are trying to do is that, what if we have a magnetic system that can do almost the same thing? The counterpart of the FRET, maybe STAT or optogenetics. So these are things that we have worked on for the past few years. And then today I'm going to tell you a little bit about magnetogenetics. And this can be quite useful for deep tissue penetration with a non-invasive remote modulation of neural behaviors. So this is a magnetic fields that you can see in the movie. This is a Magneto. So this is the Marvel Comics. And there is a character that this guy is the guy who just controls almost everything, right? And he uses a magnetic field, and that's why his name is Magneto, right? So we all know that. And then this is a long-standing question that people have interested in. And then here we are in Seoul, and in East Coast there is a river called Namdae, and then people usually let out the salmon fry, and then they travel about 30 to 40,000 kilometers over 3 to 4 years, and they come back, and the homing rate is about 10%. And then some researchers in New Zealand, they found that in the forehead of a salmon there may be some magnetoreceptors, and then it comes with magnetic nanoparticles about 50 nanometers in the form of the chains and so on. So, and then now people wondered what are the guiding principles of the, for example, migratory animals, and people occasionally found that some magnetic nanoparticles in their brain and so on. But it's not really conclusive. There are many other theories, quantum optical guiding system and so on. So we don't know. But one thing for sure is that the magnetic nanoparticle can be some kind of a neuro transducer in some way, and the question is then here we have nanomaterials and then we have a biosystem. So then are there any language that nature has developed between nano and biosystems? But people have found that one is the light-sensitive organism, that's opsins. So people have utilized that for the optogenetics. And then maybe a magnetogenetic of the receptors. So there are some, but not that many. And then can the magnetic material serve as a translator? That means that, pressure, photon, or heat, and some of those can be utilized. Is that possible or not? Maybe so. So that's something that we have initiated our project. So this is we all know that brain is activated by the ion channel gating processes, right? So we have a neuron, and then you have a cell membrane. Let's say you have a certain receptor or ion channels that can be gated by the magnetic nanoparticles. Then that's going to be the something called magnetogenetics. And then we need a mechanical force about 5 piconewton to open the gate. And this is the force scale of biology, and we have a mosquito on your skin that's 25 micronewton. It's not that heavy, but it's quite light in some way. But if you go into the antibody antigen, that's about 1 nanonewton, and ion channel gating requires about 1 to 10 piconewton in general, and then cell membrane, the movement requires much less force. And here, the chemical bonding is it ranges from nanonewton for covalent bonding, which is regarded quite strong, and then hydrogen bonding is a few piconewton range. So people now have been interested in using naturally occurring iron oxide as a cue for the magnetogenetics. So let's say you have iron oxide nanoparticle and then ferritin, and you have a pulling force. And so some neuroscientists, they utilize and they claim that you can really do the nervous control by genetically synthesized iron oxide nanoparticle. And then there are some backlashes and coming out later. And there are a couple of papers published in Nature Neuroscience that you cannot really reproduce this result. The claim cannot be reproduced by other scientists. And then there are reasons, and because the core material is antiferromagnetic, that's the ferrihydride, and the pulling force can be sub attonewton. That means that it's 10,000 times smaller than the thermal energy. So in other words, the magnetic nanoparticle is just rotating. It doesn't do much. It's not magnetic anymore. So I mean, there are many arguments that this cannot be well done. So our approach was about using some, the torque. Li Jiang just mentioned about that a little bit. So because you have a geomagnetic field and you have the compass, and then it will rotate and it will generate torque depending on the direction of the magnetic fields. So what we have done is that we first thought about design a nanomaterials that can be able to generate the rotational force torque, and the other one is the you need some material other than the ferritin iron oxide to have enough magnetic field, and then we also need a ion channel to really prove that it is only driven by the mechanical force, nothing else. So you need to design a certain mechanically driven ion channel. And then let me tell you how we have developed this. First one is the M-torque, because we just named it because it generated torque force. So you need to have a magnetic nanoparticle that has a very high magnetization value per particle. So let's say you have a single nanoparticle. Let's say we have a octahedron iron oxide nanoparticle with a size of let's say 15 nanometer or 25 nanometers. It has a relatively high magnetization value, but still that's not enough. So what we have done is we clustered them into a about 500 nanometer in size, and then just utilize that as a single magnetic probe. So that's how you can generate about 2.1 pico EMU per particle. So that's about about 500 times higher MS value in comparison to the single magnetic nanoparticles. That's one part, and the other one is you need to generate a rotational magnetic field to have a torque. So let's say you have a two-magnet system. This is the blue is the magnetic field about 20 millitesla. So the blue, and the red is a high magnetization value. So near the magnet, of course you have a gradient field, and that's very high magnetic field, and it will go down. It will go down, and then at certain distance, there is a plateau. Okay, so the plateau is about, let's say here is about tens of millitesla here. So then you pull them magnet out, and then it will diminish the overlap. And then once it reaches, let's say 16 centimeter, there is no magnetic field error, right? So, but one other trick that you can have is you have another pore, magnetic pore. Let's say 4 magnetic pores, then you can generate a enhanced magnetism, and so on. So what we have done is that we have let's say 6 magnets and then in certain arrangement in terms of the position, then you can have a relatively constant magnetic field in most of the region. So this is what you see here. And then now what we do is that this is we utilize a permanent magnet, 1 Tesla, that you can easily buy. And then you rotate it, and then this is a M2 core, and we put a fluorescence tag, and then indeed you can rotate it, the magnetic nanoparticle, by using the CMA, circular magnetic array. It's just a rotating, mechanical, not electromagnetic system. So it's simple. So now you need the third component, which is the where can we find the mechanosensitive ion channel? And then it is we all know about the 5 senses, right? But the sixth sense is defined as this is intuitive faculty giving the awareness not explicable in terms of normal perception. In other words, we have 5 senses, but beyond that is called the sixth sense. That's simple, right? So, and then something is related with proprioception, balance, and blood flow regulation, bone and muscle movement, and so on. So let's say you walk in the without even seeing anything, you can have a balance. It's because of the this proprioception and so on. And then some scientists at Scripps and other institutions, especially the Adam Potapushian, and he won Nobel Prize a few weeks ago, right? It's about a month ago in physiology or medicine. And but just finding out interesting mechanosensitive ion channels, and one is called the piezo 1 and 2, and it is about 900 kilodalton protein. It has 3 blades, and then when you pull the ion channel, that it will just open. It's amazing nano structure for controlling the ion flux. So what we have done is that we first have a normal neuron, and then it's clean, and then you need to transfect the gene to express piezo 1. Okay, so this requires genetic engineering. And then now these neurons are expressed with the piezo 1 with a tag, we call it the MIC tag. And then this is the M2 core that I have discussed. It comes with antibody. This antibody will bind into the MIC tag of the piezo 1. And then we apply the magnet, circular magnetic field. And then the signal propagation goes in. And that's what we hoped at the beginning. And then indeed, this is the neural activation that we have done. So this is the mouse, and then this is a CMA. This is circular magnetic array. It rotates about about 0.5 hertz. It's very slow movement. It goes counterclockwise, clockwise, and then as you do it, and then we can clearly see there is a temporal activation of neurons. Whenever we make the magnetic field application, then we see the strong fluorescence signal in the neuron. In other words, there's a calcium influx is clear. And the next step was about do we really replicate this in live animal? So what we have done is that we targeted the motor cortex, and here some motor cortex are well studied in optogenetics. That's why we chose to do that. And so we have a motor cortex region, and then we injected M2 core, and then before do this, we need to express the piezo 1. So we first do the viral injection and a certain coordinate of the brain, and then we injected the M2 core. And then this is the activating neurons with the C4, so we can clearly see that only where the mTORs injected, the neurons are being activated. So this is the live animal study. So we have a this is a mouse. Well, it comes with this mouse has a brain with a PH1 expressed already, but we didn't. And then also it was injected with mTOR, and then we apply the magnetic field, and so as soon as you turn on the magnetic field, then in other words, the CMA, there is circular magnetic field, and then it is rotating in the counterclockwise because we injected in the right hemisphere over the motor cortex region. So that means that your left legs are being promoted, and then you have a counterclockwise movement. And then you can do the same thing in the left hemisphere, and then now it is moving into the clockwise fashion. So that means that you can indeed control the ion channel gating in the neuron in the live animal by using the magnetic field application. So this is a video clip to summarize what I have told you. So here is a PH1 ion channel, and then you inject the mTOR, and then you apply the CMA, the circular magnetic application, magnetic application, and then it will just start the activation of neurons and then brain activation and so on. So that's what we have done. So today I have discussed about, here you have a magnetic nanomaterials, and here is a biosystem, and then so my question was, can we really interface material into biological system or not? I mean, can you really do that? It's not about macro scale or larger scale system. It's just do we really have a some language that you can develop so that biological system can understand and do some actions? And indeed, it is possible. So especially the magnetism can be a beautiful translator. It really does so. And then I think that the magnetic reception is possible with the neuroscience, and something like an mTOR that we have developed is still in premature stage, but it could be a one of the useful and effective tool for understanding about brain function and disease. And here are some neural stimulation program that people have developed. The first one was about the electrical field. We have a nano electrode and Utah probe and Michigan probes, and you just inject into, for example, some of the regions, for example, with the people with Parkinson's disease, and then you just have an electrical shock, and then it does quite good, effective outcome out of it. And then later on, people have started to utilize the light photons to activate the neurons by using the opsins, light-sensitive ion channels. And then here, as you can see, that it comes with optical fibers and so on. It's beautifully done. You can do temporal studies and almost any interesting optics-based studies, imagings, and neuromodulations. And then now I think we can have another modality that's magnetic fields. And then you can, as long as you have a well-defined bona fide ion channels like PH1, 2, and some other ion channels, then you can do remote and wireless activation and deactivation of a neuronal system. So that's what we are interested in. And then I think we have seen some some new possibilities And I mentioned about the motor cortex activation, but basically what you can do is that you can do remote control brain activity from a single cell, the in vitro level, to animal whole body system. And I think you can have a very simple instrument, because all you have is just array of multiple magnets, and then you just have a motor to rotate it in a very slow speed, 1 or 2 hertz or sub 0.5 hertz and so on. And then you can really activate the neural system. And then we think that this could be serve as a new platform for the brain research, because we can have a specific neuron activation and deactivation. And the advantage is that you can have a deep brain stimulation, for example, and you can target any part of the brains, and maybe you can also study in peripheral system too. So it can be widely used for many different studies. So I think with that, let me close, and I have multiple collaborators, and then I should thank my funding agency, and also I'd like to thank all the distinguished guest speakers to join this IBS conference. Thank you so much.
Thank you very much, Professor Chun. And then I think we can have a few questions. Yes. Okay, Professor Fisher.
Yeah, thank you very much. Very interesting. I have sort of two related questions. One is the M torque seems to be a spherical particle, yet you need to exert torque. So how do you get anisotropy in a system like this?
Right.
And the second one is, I was wondering if in your study you've done on-off type studies rather than continuous stimulation, and whether you can then basically show that the response is in time with magnetic.
Okay. So the first one is the particle that we have realized is not super paramagnetic. It has a very weak magnetic coercivity, so that it is you can regard it as a ferromagnetic, but it's almost like a super paramagnetic. So that we try to diminish the interaction between the M torque, which is about 500 nanometer interaction. So that it can be well dispersed without any aggregation and so on. So, but still, it is so that we can call it a probably the smallest nanocampus. So it has very weak. So the way that we utilize the magnetic nanoparticle, the assembly of nanoparticles, it retains the magnetic coercivity as the starting nanoparticles in terms of the torque, M torque. So it maintains and just shoot up the MS value as a whole for particle. And the second question is that you say that you have a neural stimulation and you stop, and then let's see how it goes, right? Indeed, you can. We didn't show it to you, but it only activates when the CMA magnetic field is applied, and then once it is gone, it just stays as normal.
Okay, so Professor Che.
Yeah, thank you for the wonderful talk. Very interesting. I have two questions. One is you show how to control the motor cortex in the mouse, and then you control the behavior. So that's very interesting. I think you also mentioned a little bit, but I would like to know what's your further plan for the neuroscience to apply this technique for neuroscience.
Yeah.
The second question is, I think you answered several times, and I also asked the same question to many different people. So what's your comment about the material biocompatibility, or how to remove it from the body after treatment?
Okay.
Thank you very much.
Right. So the first, I mean, you can do a lot of interesting study, because you can. Is it like there is a gate, and then you open and close, then you activate some cellular signals, and it includes the neural activations. So people have studies. One thing that you need to do is whether you can here we activate the neurons, but can you do inhibitory, ion channel gating? That means that you depress the action. That's the one thing we haven't done yet. So that's something that we are working on. And the other one is you need to have a certain targeted delivery of, for example, mTOR. And that requires, that means that the specific neuron targeting activity is very important. So there are, it's more into the bioengineering aspect than the materials design. But we are working, because I have my collaborators who are developing many different types of biotechnology tools to have a high specificity of the target, and also the activation and deactivation of certain neurons. And the second question is about the iron oxide, because we utilize iron oxide, and the delivery, the amount of iron oxide is very low to perform this kind of capabilities. So we have done many biocompatibility studies. As long as you use, for example, Fe 2 or 3, that means the oxidation is 3, and then if the dosage level is very low, it's perfectly fine with your body. So we are not worried about the side effects of the nanoparticles, because the dosage level is extremely low. So for this study, I don't think any potential side effects, if any.
Okay, thank you very much, and then please join me. I'll give him a big hand, Professor Chan.
Thank you.
Yeah, thank you. So I think that the next speaker, we need to move on, and then Professor Per Fischer, and he is a professor of physical chemistry at the University of Stuttgart, and he has the independent micro, nano, and molecular systems lab at the Max Planck Institute for Intelligent System in Stuttgart. So he received an Attract Award from the Fraunhofer Society, and in 2016 he won a World Technology Award, and he also received the Steinhofer Prize in 2016 from the University of Freiburg. So today he will give a talk named as the active delivery of therapeutics and biocompatible magnetic materials for nanorobotics. Please join me. Welcome Professor Fischer.
Thank you. Thank you, all the organizers. Thank you, Professor John. Very nice to be here. So from Stuttgart, Max Planck and the university, and I'd like to talk about aspects that are related to active delivery, and in particular with a focus on nanosystems. So before I start, the thanks goes to the current postdocs and PhD students as well as alumni whose work I'm presenting here, and of course we have several funding agencies. In this talk, because I guess it's IBS for nanomedicine and it's nanomachines, I want to emphasize the aspect of nano, so really nano in the sense of nano being smaller than micron, and why does that is important, and in fact I think in some cases actually essential, and how it relates to magnetic systems as well as chemical systems. And so I want to start with the magnetic part, and I will give a little motivation, but I'll be fast and talk a little bit about our work that we've done for several years, and then focus on some current questions. So one motivation that lots of people of course have, and it's been already implied in the previous talks, is that you don't want to in certain cases be systemic in your application, but you'd rather like to be targeted, take something to a very specific location. And another challenge that you might have is that very often things are not able to reach certain regions. We know of the blood-brain barrier, we know of the skin, etc. These are barriers that prevent the distribution of pharmaceuticals. So if you just take things and inject them in the blood, or you take them orally, there are of course natural ways how to distribute them. The difficult part is to reach certain regions, and we can take inspiration, and I think this is kind of fascinating, that nature has these very unusual systems. This is now not quite nano, this is hundreds of microns. It's things that look very strange because they need to break symmetry and they swim in water, but even more remarkably, when they reach the skin, they can drill in effectively in very short periods of time. Something that absolutely nothing can do in medicine or pharmaceuticals. In fact, most things that are skincare products will never ever make it. Even a molecule like DMSO is the only one that is well known to reach deep down, and most things don't make it. So here is a soft thing, 100 microns, fully driven by chemistry, and it's able to do this. So that's kind of remarkable and fascinating to ask how to do it. Just a few intro words, because we've had lots of things about magnetic particles, and there have been studies. So one thought might be that you take magnetic particles and you locate them, for instance, on the outside on a tumor. And there have been early studies, actually successful studies, tumors in cats, and they have been very good in that you can inject in the blood, you can localize near the tumor regions, you get higher effectivity than if you don't do this. But there are sort of geometrical constraints by the virtue of these gradients. Number one is they're typically weak if you're dealing with a real body. And number two, you always pull to the outside in some ways. So that limits your application space. These studies actually were quite successful early on, but then they were dropped. And I don't exactly know why. But the bottom line is that for applications within the body, it's a little tricky for these magnetic reasons that I've mentioned. And so for several years now, we've looked at this method of being inspired by sort of the locomotion of bacteria. And everybody seems to show this movie here, which is from Linda Turner and Howard Burke. I was actually in the same institute as Howard Burke. That's why I got interested in this whole thing. And the application is that you can now turn this locomotion of this corkscrew type mechanism by looking at a torque. And we just had a lovely talk on torque, and in fact Li Zhang and Brad Nelson have also developed fascinating systems that are based on torque rather than gradient pulling. And the advantage is that you can have a homogeneous field, and this is easier to establish, and therefore you're not limited to the outside, and you can direct things, and you can in general exert much larger forces. And so for those that are not familiar with this, the idea is very simple. You take a screw, you glue a magnet to it, you rotate it, and then it moves. And the field is old. This is from 2000, Japanese group that has used a very large screw to drill through actually beef here. And the difficulty, of course, is the torque depends on the magnetic material. So as we go small with the cube, the volume goes down, the magnetization goes down, and that's typically a difficult thing. So how can we still use small materials and we can nevertheless benefit from this kind of mechanism? And one area of interest that we've had for quite some time is that tissues are very interesting structures. They are typically, we've heard hydrogels play a role. These tissues, they are macromolecular, they form a network. And so if you have a limited amount of force, one strategy is that you could be small enough so that you can sneak through these networks here, which means you can actuate things more easily. Now, of course, you also reduce the magnetic moment, but it also has the advantage that you become more mobile, and the alternative is to make the structure larger and larger, and you kind of basically have to push your way through or destroy this tissue. And so how do you measure this? You can take particles and you can just drag them through a tissue, and then you can measure, and we've done this for several systems, and here's one representative one. So if you have micron-sized particles, typically you cannot move them in most fluid-like or tissue-like systems. But if they get smaller, typically hundreds of nanometers, then here the velocity picks up because it becomes easier to navigate. And now we've already talked about magnetic particles. We like to use physical methods to make them because we have excellent control over the material composition. We can do shape. We can do the combination of multiple materials and functionalities in one growth system. And we use a method whereby we use a shadow growth. So we basically shadow deposit onto a pattern substrate. And the patterns are done very quickly. Basically by spin coating, we get patterns of nanoparticles. And on these nanoparticles, we can grow these structures. So we can make on a 2-inch wafer about 100 billion structures, and they are typically done in a few hours. And so this is early work, how we started. These are colloidal structures. They are a little bit bigger now, length scale of a bacterial cell, and one particular shape that we can make very well are these screw propellers. And here they are in water, and if you fluorescently label them, you can navigate them very nicely in water. And so that, of course, brings the question, can we take this kind of thing and can we examine tissue-like structures? And so we take the wafer, we release these propellers into solution, and then we can have controlled size, shape, surface chemistry, and then we can study how we can navigate through various tissues. And by this method, we're also not constrained to the living world. So the living world are bacteria, and the smallest swimming objects, in my opinion, that are actively swimming are about 800 nanometers in size, or something like 500 nanometers. And then nature doesn't seem to swim actively anymore because it doesn't seem to make much sense. Diffusion does the trick. You don't want to waste energy because you're anyway not going very far. But making artificial structures, we of course are not a living creature. Therefore we can make things, if this is a bacterial cell, we can make them lots smaller. And we can then explore this diameter here so that we can be small enough to make it through various networks. In this particular case, the magnetic part is this bright bit here. This structure has a diameter of 70 nanometers. So therefore we can really navigate very small networks. And now I want to talk about two aspects. We can make lots of them. We can actuate them in water. They're very small. They can go through tissue-like structures. Can we go through real biological tissue, deep, rather than being in cavities? And then I want to address this question of the biocompatibility. And so again, briefly, two quick examples. One was the mucus, and then it's interesting that even nature, the pathogens, can have problems to cross barriers, and that you really need to combine multiple things. You often need to combine the mechanical actuation with something. In this particular case, it actually needs a biochemistry. And so quickly, this is a study we did on mucus and bacteria. Helicobacter pylori, Joe Wang mentioned it earlier on, is something that can give you a stomach ulcer. So the bacterium goes through the mucus, exposes the wall, and then you can get, at the low pH, you can get inflammation. But what fascinated us is, this is a PNS study out of Boston. If you look at one single bacterium, it's actually, even though it's supposed to swim and drill through mucus, it's not able to go. It's kind of spinning its flagellum, but it's not propelling. But somehow we know it needs to go through because it's able to infect the stomach wall. And if you zoom out, you see these little guys after some time swimming very nicely. And that is because the bacterium actually does multiple tricks. It not just has the mechanical force, it also has a chemical helper. It excretes enzymes, urease, urea converted to ammonia. Ammonia increases the pH. And by the way, I've had some very nice Korean food. And if you eat, I probably cannot pronounce it, hwang ho ho, then you will have some ammonia taste where exactly the same mechanism happens because sharks and rays excrete uric acid into the blood, and if you decompose it, you get this ammonia. But got nothing to do with my talk. So let's back on track. If you then look at the mucus, you have a fairly high viscosity at low pH, pH 2 or 3. You add this ammonia, which the urease does, and then you can see here 3 orders of magnitude drop in viscosity, which means it can swim. And we can mimic this kind of thing very nicely. Here are propellers without enzyme, and here they are with, and then they will navigate very nicely through. So we can combine the mechanics with some biochemistry to allow propulsion. Now, the last one that I'm going to go on things that we have done a little while ago is by also looking at the surface chemistry here, because adhesion can play a role. And this was a study where we wanted to go through the vitreous of the eye of a young eye with an intact vitreous for a long distance. And the long-term goal is to maybe be able to do things like gene therapy. I develop genetic material and bring it to the retinal cells. And again, you have this problem if you go deep down. This is inside the vitreous on a young preserved eye, so that it isn't already decomposing. There is always a tricky part that things get adhered very easily and they don't move. And in order to overcome this problem, Jigwan Wu kind of borrowed this idea of this coating, and this is a movie that was done in the by the science people who illustrate what happened. Here they are with the coating. You can see these structures navigating very nicely, and the coating actually is one that is first a silane, and onto the silane there's actually a perfluorocarbon liquid. So it has a liquid coating on every particle, and the tricky part is how to do this without having them all adhere, which Ziggy Wu managed. And then we not only looked in the microscope, but we actually then also did this system, and we tried to use, as is done here, actually the instrument the surgeons use, which is optical coherence tomography. And we can see how this whole group of particles reaches the retinal cell with very good control and focusing. So now I'd like to get to the bit which addresses this conundrum that we have been struggling with for a while, which are these magnetic particles, and the fact that they need to be biocompatible, and that often we don't have enough force. So the magnetic systems tend to be very cumbersome and bulky, and I think Brad Nelson will probably talk about some very nice technologies that show how we can get strong magnetic fields. But nevertheless, you want to have maximum torque, so you can work hard on your electromagnets or magnets, or you can try and improve your magnetic material. And at the same time, you want to have super strong magnets, and they should be biocompatible. And if you look at the list of magnetic materials, iron is corrosive, nickel and cobalt is toxic, iron oxide is okay, but doesn't have such a great high remnants in its magnetic field. And you would really like to have the best magnets, which are these neodymium iron boron ones. And there seems to be this wonder material, at least I think it is, which is this iron platinum in the L10 phase, which are FCT, where you see iron and platinum, which really ticks all the boxes. It doesn't corrode, it's in many studies been shown to be fully biocompatible, and it has really incredibly high magnetization. Early work looked at synthesis. So this is a paper from 2000 by some well-known names that did iron platinum nanoparticles. And it really is nice because it shows this very large magnetocrystalline anisotropy. But in synthesis, they managed to go up to 10 nanometers, and the domain can potentially be larger, so it would be beneficial to go larger. And there is this one tricky step. You've got to go from FCC to FCT, which then means here you have to get rid of the solvent and you do some annealing step. Now, we don't need to do this if we immediately go to the vacuum. We can immediately be in vacuum and easily do annealing. This is a paper from our institute with Gisela Schütz, and actually my student who is kind of in the audience, Wang Ho Zhong, he's now assistant professor at GIST, was involved in this, looking at this wonderful property of this iron platinum L10. It has a coercive field of 7 Tesla, right? The energy product is something like 50 megagauss Ørsted and a very high Curie temperature. And at the same time, it's all biocompatible. Very nice energy product. And by playing some tricks, you can get the magnetization even to exceed that of neodymium iron boron, right? This is really remarkable. If you do vacuum type processing, the magnetization will be determined by the substrate, i.e. lattice match. It will be determined by the deposition temperature, it will be determined by the film thickness, and you can even increase the magnetization further if you, for instance, give it a gold coating. So this is kind of remarkable. And if you look at the binary alloy phase diagram out of this publication here, then we need to be roughly in the 40–50% iron platinum, which means it's also conveniently targeted with co-deposition and other tricks. So this is what my PhD student Vincent Kadiri did. We grew these propellers like I described, and then we used a little bit of a deposition of a thin film on top, and actually reducing the material can increase your magnetization, which is another crazy thing. And then a small annealing step from there, and you have then this fully biocompatible, very strong magnet. Then he used the polyethylene amine coating and added some plasmid DNA to prove that it's working. Here is this black curve, is this wonderful magnetization curve, really, really superb, compared to iron, compared to nickel. It's just wonderful. And he basically transfected some lung carcinoma cells to show that we can even get this GFP in, and that the cells are happy, and the usual tests that the biologists do. So I think this is promising. And I want to wrap up this section because I also want to talk something about chemical systems. So I think this is interesting for this field. It's a very promising magnetic system. You can use various fabrication techniques to get very large numbers. As we've shown, there is a need for nano. Sub-micron can really facilitate penetration. You can functionalize these things, and you can add this magnetic control. And the benefit is then you probably can get away with slightly simpler external magnetic actuation systems. So, second part. See, I still have some minutes. I'm a bit worried here that I need to rush, but I want to talk about chemical. So one is we have these magnetic systems to drive nano systems to various places, but what about chemical? And there are two aspects I want to talk about. One is the locomotion and propulsion, again with a focus on nano, and the other one is just the fact that these things are catalytically active. And so this relates to the field of active matter or chemical motors, and I think we have seen many examples by Joe Wang. The mechanism is that the reaction causes locally a concentration gradient across the particle. The concentration gradient across the particle causes a flow, and the flow is non-trivial actually. It's also non-trivial to predict the direction of the flow, but the particle will then move because there is this concentration gradient and there are flows. This is what is known as a chemical nanomotor, chemical motor, active matter, etc. Or self-phoretic or swimming particle. And so, with a view to nano, the question is, we have seen these examples that large particles can do this. We can either drive them magnetically, or we can drive them chemically, or we have biological swimmers. They are all in the micron scale, or very close to it, just under. Can we take these ideas and make our liposomes active? Can they swim? Right? COVID, corona drugs, liposomes. They are active and they are fantastic in the sense that they work, but they're not actively moving, propelling. Can this even work for enzymes? Can this work for even smaller structures? Do these concepts scale down or not? I think this is a very important question because there have been publications that claim that even enzymes can move actively, i.e. they swim more when they are doing reactions, and molecules, even molecules are supposed to do this. If this were true, it would really change the ball game of trying to think about pharmaceutical or other things, because now you could be implementing this in small molecular systems. And the whole field probably started with observations based on fluorescence spectroscopy. This is urease, and the first claims was that during the catalytic cycle, the urease does something like a non-reciprocal shape change, which is like a sort of Purcell type swimmer. And then it also on fluorescence spectroscopy based, people observed that alkaline phosphatase was observing diffusion increases, almost 30, 80%, even higher. And then people started to come up with ideas of what could be going on. So basically the chemical energy release could be pushing the fluid, and that could be pushing the enzyme. And more papers, right? Catalytic enzyme, active matter, enzymes leap, and then even that in reactions, the catalysts are starting to move. So quite exciting, and of course we wanted to get into this and look at it. There could be interesting phenomena, i.e. new physics, although this is a little bit questionable. It makes little sense in terms of what we know. Or it could be boring, right? We can just be heating the solution, that's boring. Or we could have things falling apart, becoming smaller, and because they become smaller, they move more. That's also not interesting. Anyway, it's an interesting question. Claims have been made of ballistic, i.e. jumping motion of 10 nanometers. This is phenomenal, right? Because we know that everything is so overdamped at these length scales. Energies in proteins dissipate in picosecond timescales. You know this from ultrafast measurements. And typically proteins come to a halt, or anything comes to a halt in a very short time. So if you, Gedanken experiment could take a bacterial cell and you could switch its engine off, it would actually stop in 1 angstrom just because of the drag. Now, of course, Brownian motion will move it around. So people have looked at these kind of mechanisms theoretically. Is this possible? And Golestanian did some numbers. So the self-phoresis, i.e. using the concentration gradient to pull something, that prediction is too small by 15 orders of magnitude. Kinetic energy, i.e. could we be doing some impulse mechanism, too small by 4 orders of magnitude. And then stochastic means you're just sort of having the randomness help you is also too small. Heating could be something, but this is actually one of the parameters that we have very good experimental control, and we can check whether that's happening or not. So there is a big question, and we had to get into this because this is quite fundamental. And so fluorescence correlation spectroscopy is used in a lot of these measurements. You take the enzyme, you send it through the focus of the laser. There is a fluorophore on it, and it will emit light. And basically, and then you record the light. And of course, if it's very fast, it will be a short light burst. If it's slow, it will be a long one. And thereby, you can induce by correlation how quickly it's moving. But there is a problem with all of these measurements, in our opinion. And that is the diffusion, the speed of motion, if you like, is deduced from intensity measurements. Right? That's the same true for fluorescent correlation as well as NMR measurements. They actually ultimately measure intensity, see an intensity change, and say this is enhanced diffusion. But as an experimentalist, I can tell you intensities can change for a million reasons, and they can be other than diffusion. And this is the explanation, in our opinion, what went wrong in the Nature paper. And what went wrong is that you have a fluorophore, and while the enzyme is catalytically active, it will actually have a molecule, the substrate, interact with the enzyme. It's obvious. Otherwise, if it doesn't have a substrate, it wouldn't do anything. But it turns out the substrate is a fluorescence quencher. So the substrate will switch off the light, if you like. And so if you go back, and if this were going through here and the light would go off, then you would, of course, record a shorter pulse duration of your light, not because it's gone through faster, but because the light has gone off. And so the shorter light burst is then interpreted as a diffusion enhancement, but it's actually just a mechanism of fluorescence quenching. And we've simulated this here. And here's our control experiment, and we've published this in this accounts paper. And here are basically the time of the correlation. The green is a very small molecule, rhodamine. The black is the enzyme. So the black is below the green, hence it's slower. Now you feed this, the substrate, which they have done in the paper, and the substrate is a fluorescence quencher. So it now looks like it's going much, much faster, right? But it isn't going faster, it's just being quenched. Because if we replace the substrate with one, that doesn't quench, we still get the same result as if we'd done nothing. So we don't think there's any genuine result there. And we've looked at other enzymes, and we've also recently looked at reports that have claimed that in catalytic reactions you can get enhanced diffusion. And this is a comment we wrote early this year, and now we have a paper coming out in JACS where we look very, very carefully at the NMR measurement. And again, there are many reasons why your intensity can go down in the measurement, things like paramagnetic ions, etc. And so our conclusion is, other people don't agree with us, our conclusion is enzymes and molecules don't show measurable enhanced diffusion, which again actually couples back, which means if you want to use this effect, you will have to use nanoparticles. And I think that makes sense because proteins in nature, if you look at the rotational constant, is incredible. They're so small, they rotate so fast, so they walk on tracks or they sit in membranes. They're localized, right? If you were to create a concentration gradient and you're spinning super fast, you can't maintain it. And so either you slow it down by putting it on a bigger structure or you anchor yourself. So that's our conclusion. And I think the chairman will give me trouble now. 2 more slides. I just want to say, bring this chemical story to a conclusion. There's still a very interesting possibility because these things are active, maybe not in the propulsion sense, but they're active in the chemical reactive sense. And so we've already seen that things like urease can facilitate propulsion because it changes the local environment. And so very quickly, 2 slides and I'm done. How can we make highly reactive enzymatic systems? So we worked, Dirk Rotenstein and Joachim Bill, we looked at bacteriophages, filamental viruses, and you can anchor enzymes on them. So these are very thin nanowires, 6 nanometers. You can genetically modify the peptides, and then you can decorate them with lots of enzymes. So enzymes typically and often don't like to be on inorganic substrates. They often show reduced activity. They are super happy here because it's proteins and it's very thin. You don't have a problem with diffusion of substrate and product in and out. And so you can make all sorts of interesting systems where you bring these enzymes together and decorate your system. And just to end up here, to prove that the catalytically active enzyme has a real effect on solution, we used urease on bacteriophages and we put them together in a little chamber, and then we took tracer particles, and the reaction is actually in blood. This is done, a little bit of urease in blood. The reaction causes density gradients because it has reaction products, and then you can get convection flows. And this is just to show that maybe the swimming idea at this enzyme level is something we should look at carefully, and we don't believe it's there. But you still have fairly big effects if you take these structures and you put them in solution, because they will have large effects on the hydrodynamics via other mechanisms, which is either chemical gradients or density gradients. And so the bell went. My conclusion is, in our opinion, and we're convinced there are artifacts and mistakes in lots of papers that look at this, no self-propulsion of enzyme and molecules. You really need larger nanoparticles, and I think that makes sense. It's also what we would expect from physical chemistry. But nevertheless, you can pump fluids and or you can use it to facilitate the penetration of barriers. And very finally, there are papers on chemotaxis. And this is again complicated because people are muddling the nomenclature. They're taking things which are just diffusion and call it chemotaxis. And it's also non-trivial, because if you do this on a piece of paper, you will conclude that you cannot get chemotaxis, you can only get anti-chemotaxis. But I mean, really the work here of Michal Popescu together with Clemens, we show in this paper that if you integrate these gradients across the particle, depending on the affinity of the product molecules you create in your reaction, you can get chemotaxis or anti-chemotaxis. So in principle, these ideas work. And I think with this I should really end. So thank you for your attention.
Thank you very much, Professor Fischer. And then we can take a few questions. Okay, here. Okay, Professor Nelson.
Okay, thanks, Peter. Great talk, like always. So I guess I had always just operated on the assumption when you get to around a micron, I just see Brownian motion knocking everything around, and I didn't think further than that. The only thing I can imagine is these enzymes are changing the structure. I mean, obviously, changing the bonds and all that. Why? Where is it? I mean, you kind of went through it, but where are people making this argument that Brownian motion is not just a random knocking around is not the dominant issue here?
I don't know. I mean, it was sort of extrapolating from these wonderful results from the micro world, maybe, thinking that they would go down to the molecular world. I mean, I should say there have also been some nice papers. For instance, Eric Lauger has a PRL paper that if you just basically, if you were to flap your wing reciprocally, this is called stochastic motion, then you can, in principle, also go further than if you don't do this. But it's off by what has been observed. I mean, people have seen incredible increases, which is never explained by this.
It doesn't work if it's a viscoelastic material.
It doesn't have to be viscoelastic.
Not viscoelastic.
Yeah, this is by purely stochastic mechanisms. But yeah, I don't know where this idea has come from, and there have been several explanations, but I think it's probably been driven by experiment, that the experiment has shown these large changes in intensity, which were then interpreted as diffusion increases. And then, I mean, the paper by Bustamante in Nature was claiming chemoacoustic effect, that is basically the energy release pushes onto the solvent. These are things which are, yeah, not really what one would expect from a sort of small molecule approach. Yeah.
It reminds me of when Robert Brown first saw Brownian motion. he saw pollen with pollen grains,
yeah.
and he thought it was the life force, right?
Yeah.
I mean, these guys are. It's like, oh, it's an experimental result. Only then he looked at pieces of quartz powder,
where he saw the same thing.
and he saw the same thing. So these guys should do another experiment.
Well, I think it's an interesting idea, and of course one thing that I should say is that all of this is non-equilibrium, so we're not at equilibrium, and so all the theories become rather difficult, and so there's always scope for surprise, and I think it's good to look at this, and it's in principle possible, but I think it wouldn't be to such a large extent, at least from what we looked at it.
Okay, so there's one more question back there from the student.
Thank you for your great talk. I'm Jung Soo-la from IBS CNM. So through your research, you've demonstrated that you have a good fabrication method for making these nanostructures, and also you've demonstrated the material advantage of iron platinum as a hard magnet. But so my question is it possible to operate these iron platinum deposited nanostructures in a swarm without causing aggregation, because iron platinum is it will have a magnetization at high fields?
Yeah. I mean, this would be a general answer. Will apply to all magnetic materials. I mean, if you make sure that they keep at a distance by adding a coating, or you add some other material, you can really prevent aggregation. It's correct. The more magnetic you make it, the more likely it's likely to aggregate. But we are putting there something like a 40 nanometer magnetic structure on something that is several hundreds of nanometers. So these things are not allowed to come together that strongly to really truly form aggregates. And by the way, that's a holy grail for all the students. If you can grow magnets like this on a large scale, I think you can retire very quickly, because you could replace the rare earths. And that's a conundrum. It's actually difficult to grow very large magnets with these systems, and that's where the nanoscale wins, where you can do so much better.
Thank you.
Okay, thank you very much, Professor Fisher, and then let's give him a big hand. So I think this is the end of the morning session. Before we going out for lunch, I think that we need to take a group photo. So can you? So yeah.
Okay, let's start the afternoon section today. So thank you very much for your visit here. And so next section, in the first speaker is Professor David Gracias at Johns Hopkins University. And the title is Physiologically Responsible Shape Change Devices for Drug Delivery and Surgery. Yeah, let's welcome Professor Gracias. Okay.
I would like to thank the organizers for inviting me to this exciting conference. Is it blurry a bit? Is it okay?
Yeah, it's okay.
Yeah. Okay. So it looks blurred, right?
Oh, really?
Yeah, the fonts. It should be crisp.
Yeah, okay, so let's wait. Yeah. So she will turn off the light.
No, but the problem is with the font. You see the font?
The focus is not.
Yeah. Okay, so today I'm going to talk to you about a little bit different from the other talks about shape change devices. And essentially, what you're seeing is a video of a 1 millimeter glass capillary. And there's a gripper going inside. It's being moved by a magnet, and it's triggered by temperature, and it's strong enough to bring out tissue. I need to have a conflict of interest statement because some of the patents related to this work are in license agreements, and I have other patents. And I would like to acknowledge the funding from the NSF and the US National Institutes of Health. Essentially, we wrote a paper. I don't know why the fonts aren't very clear on this thing. I hope you can read. Essentially, in this paper in Nano Today in 2020, we talked about this era of active matter therapeutics. So if you look at the history of medicine, especially in drug delivery in the 1950s, mainly it was injections and powders. And then we moved into liposomes. So for example, in the US, for chemotherapy, we use liposomes for virus vaccines. And these are nano and microparticles. And then they became very fancy. So we have particles with receptors on them, needles, etc. And we are in this era now of, you know, things that move by bubbles, etc. And in the future, I believe that we will also have more machine-like structures which actually can change shape. And so you'll see a futuristic vision of some kind of robot that changes shape, you know. And today in my talk, I will argue, you know, that we have made, you know, many advances in this, in enabling this era. Okay, so if you want to put this in perspective, my clinical friends at Johns Hopkins Medicine, they tell me that they already use the pill cam. So it's already used in the patient routinely right now. It's about 1 centimeter scale. You can see the pill cam. I would call this heavily integrated. So on the one side, you have electronics. You pack it small and small, and you make a pill cam. On the other extreme, and this is a paper I did with Samuel Sanchez and Oliver Schmidt, you have extremely simple shapes, like this is a tube, and it moves. And what I'll talk to you today is something in the middle. So it's a bit more integrated than this tube in the sense that it has a lithography, a lot of lithography, and the size is a bit larger, but it changes shape. So I would put it somewhere in the middle. And really our vision is to move towards putting electronics towards heavily integrated small devices that change shape. And you know, many of you know this quote, so I will read it though. This is a quote from Richard Feynman's famous lecture. He says, "It would be interesting in surgery," a friend of his told him, "if you could swallow the surgeon." So this is not a new idea. People all the way back, this talk is from 1960s, 1959. This idea has been put forward. Okay, so my inspiration for my device is this cell. So the macrophage. I think what we are trying to do in our lab is to integrate, you know, biosensing. So the way this cell works, there's chemotaxis and other things, but essentially, you know, it chases down pathogens, and there are antibodies, and it can bind to the pathogen, and it can gobble it up. And you know, it's my dream to make a synthetic macrophage, to make a machine that we can put in the body, and it can sense something, either a tumor or something, and it can do a functional task. I think 4 main elements need to be achieved. One is biosensing, you know, shape change, translation, and safety. Is there a time? Yeah. Okay, so the first innovation we made is a very simple idea. Okay, and it's the idea of a micro spring. Okay, so if you take 2 thin films, and we often use chromium and gold, and you deposit them on top of each other and you release them from a wafer, so we use lithography, photolithography, they will bend spontaneously. Essentially, this is like a spring. One of the layers we make in tension, so it's like it's stretched, and the other one is in neutral stress. So effectively there is tension, and when you release it, when it's on a substrate, it stays flat. When you release it, it bends. Now, our innovation was that we thought about what if we put a 3rd layer on top of it. Okay, so we have this spring, it wants to bend, but we put a 3rd layer, and this is what we call a responsive trigger. So again, the advantage of this is it's extremely simple idea. You can pattern thin films with all kinds of stresses. You can make nanoscale structures with it. And you can get energy out of it. So a lot of people are trying to use batteries or, you know, non-contact energy, you know, RFID magnetic sources. But in this case, you have stored the energy in the spring. So you have the film, it's ready to go, and you just need to trigger it. Now, I won't be able to talk about the hard work we had to do in terms of mechanics, because if the trigger has to be chosen very carefully, so that when it's stiff, it arrests, it does not allow the spring to bend. Okay, so it's chosen with a lot of modeling. We do finite element modeling, etc. And we build an entire device using lithography, and these springs are in the middle. Okay, so there's a lot of lithography. We have a rigid segment, a thicker part, and in between are these joints. And then the game is basically if something will react with the trigger. Okay, so it could be temperature, it could be pH, glucose, enzymes, etc. And it will either soften the trigger or it will dissolve the trigger. So if it's an enzyme, it can dissolve the trigger and it will release the spring. In a way, it is like a mousetrap. Okay, so it's a one-way device. You store a lot of energy, and when you release the trigger, it's like a bullet. The gun goes. What is interesting is that the fonts are not right. This should be micronewtons, so there's something. It can release, not millinewtons, micronewtons of force per hinge. And we've seen that it can actuate very fast as 67 milliseconds. So in a way, it is like a spring or a gun. Okay, so what are the advantages? We can make tons of them. This is wafer scale integration, so we can make tens of thousands, millions on a wafer. They look very small. Typically, we see them in a vial. When we go to the surgery room or the hospital, we take a vial like this. They all operate in mass. Okay, so when they hit the physiological condition, they all will close. So we don't need to actuate each one. We use wafer scale lithography, and they are small enough to capture even a single cell. So one of the questions I often get asked is, what is the right size for a nanomachine or a machine in the body? And the answer I always give is that the body, in our body we get oxygen by red blood cells, so we don't get free oxygen. And it's known well that almost every part of your body has to be 100 to 200 microns away from a capillary. So almost every part of your body, the red blood cell has to reach in order to give it oxygen. And the size of a red blood cell is 5 to 8 microns. So actually, if you can make structures that are about 1 micron in principle, now of course red blood cells are squishable, and sometimes they squeeze themselves through tiny capillaries, but 1 micron is a good size to actually traverse most of the body. And of course we can make them with magnetic materials in them. So anything you can do with wafer scale lithography, you can do with the grippers. Okay, so let me spend the next few minutes telling you this is almost 10 years of work, some more recent and some older. So this is a recent paper that came out in Nano Letters, and essentially this is a single cell gripper. So again, it's a latch, so you see it coming, it's being guided by a magnetic field. And then we are actuating this by temperature. We use the food wax, a biocompatible food wax that you eat. And when you heat the wax, it closes. Okay, so it can catch a cell. This is a single cell. These are MDA, MB231 cells. It can also do a biopsy at the single cell level. So it has enough force to pull some cells out of a clump, and this is at the single cell level. These are quite hard experiments because they have to be done under a microscope. The resolution needed is high, etc. The important thing I want to emphasize is that this is a live cell. So whatever we are doing is not killing the cell. This is the evidence. We have a single cell inside the gripper, and we've stained different parts of the cell, and it's alive. So the process of capturing the cell is biocompatible. It does not kill the cell. These grippers are 70 microns when open and 15 microns when closed. And following the work of John Rogers and others, we can make them out of bioresorbable materials. Okay, so there are a lot of thin film materials that are dissolved in the body. And in this case, we are using silicon-based materials, but you can use others. You just have to choose the stress appropriately. Years ago, we showed how we can do biomolecular activation. So in this case, we took those microgrippers and we used 2 biomolecule gels. Okay, so again, this was lithography. In one case, we used gelatin. So gelatin is a gel. We developed a photo patterning process to put gelatin. And gelatin is only on the hinges. And we use another biomolecule, carboxymethyl cellulose. And so we have a gripper with 2 biomolecules as triggers. And essentially, this gripper is very sensitive to the enzyme. Okay, so if I take a gelatin micro gripper and I put it in cellulase, so cellulase only attacks carboxymethyl cellulose, then only the cellulase ones close. And if I put it in papain, which is an enzyme which degrades gelatin, then the other one closes. So you get enzyme selectivity. And we showed, that you can do autonomous kind of biopsy. So if it faces, again, think about the macrophage. In this case, it's a very simple idea. If the environment has the protease, then the gripper with the protease trigger will close. Okay, and this this paper was featured on the cover of JACS, and essentially our vision is shown here. So imagine you had a tissue and you had cells that were cancerous. And cancer cells, they often have enzymes like MMPs, okay, so matrix metalloproteases. Then the gripper near the normal tissue would not close, and the gripper near the MMP tissue would close. And in a way, it's biosensing coupled to actuation. The next few slides, I'll talk about in vivo. I want to acknowledge my long-term collaborator, Florent Celarou. This is in the division of gastroenterology. I should tell you that in vivo studies are very hard. You can always do things in a dish, but as you can see from this image, this is in a live pig. It's first of all, you cannot see well. It's dark. It's very messy. It's full of fluids. The animal is moving. So it's very challenging. If you take a pristine dish and you show some motor, it's quite different from actually doing it in a live animal. Okay, so what did we do? This paper was the first biopsy, with a sub-millimeter tool. So earlier this morning we saw the bile duct. We did this experiment in the bile duct. So the bile duct is a hard to reach place. It's also implicated in cancer. So many liver cancers start in the biliary tree of the bile duct. And so there's an urgent need to biopsy inside the bile duct. Unfortunately, the biopsy forceps cannot go in. Okay, the hole is less than a millimeter. Typical biopsy forceps look like this. So this is a clinical biopsy forceps, and our grippers are orders of magnitude smaller than the biopsy forceps. So this is a conventional biopsy forceps versus a gripper. So what do we do? This is a CT image inside the animal. You need to use endoscope. We come with catheters into the orifice of the bile duct, the opening of the bile duct, and we push the grippers in. And we wait a while. These are temperature responsive grippers. So they are introduced from a cold state. We go to the surgery room with grippers on an ice bath. And then when we introduce it into the animal is at 37. So it's autonomous. You don't need magnetic fields or anything to actuate them. They actuate, and this is a video in a colon. And you can see they're very small, and then we can catch them because if we put a magnetic film, we kind of fish them out like a fishing rod. So think about it like spraying seeds on a grass, and then you fish them out. And the important finding was that we could get good enough tissue to do, this is classic histology with H&E stain. This is cytology, that's diagnosis at the single cell level, and genetic analysis with the tissue that we got from the animal. So in principle, we were able to go into the bile duct of an animal or the colon and retrieve high enough quality tissue to do diagnostics. Recently, we use these grippers. We have introduced this concept of a therapeutic gripper. So because we can do wafer scale integration, we can do multiple layers of integration. So I would draw the analogy to the microelectronics industry. They do wafer scale, and our devices are also quite heavily integrated. We have 3-4 layers of lithography. In this case, we have the stress bilayer, we have the trigger, the temperature trigger, but we also put a patch of a gel, chitosan gel, and in the gel we put a painkiller. Okay, so the hypothesis was that we wanted to increase the residence time. So there are many patches, drug delivery patches on the body, okay, nicotine patch, etc. But on the inside of the body, it's hard to put a patch because you cannot reach the stomach. And if you just eat drug, it clears away very fast. If you take particles, they also clear because the inside of the GI tract is like a conveyor belt. It's moving and it clears the particles. We looked at the literature and we found many tapeworms. Okay, so this is a picture of a tapeworm. And these kinds of hookworms and tapeworms, they can live in your GI tract for years. Some people have for decades. So we wondered why they could live and normal drug delivery could not live. So grippers, in this case, have a drug patch. And we introduced this. This is a rat model. So we introduced the grippers through the colon. And we are trying to see residence in the colon. Okay, the important finding in the interest of time is, I cannot go through the whole paper. This is a lovely image taken by this Arijit Ghosh, who was the first author. And this paper was done in collaboration with Florent Celaru. This is post-mortem CT from the colon. So we took the colon out of the animal. And you can see the grippers going quite deep into the colon. Okay, and essentially we measured the quantity of drug in the bloodstream. And we found that compared to a control drug, there was a 10-fold higher, concentration of ketorolac, and 12 hours after administration. This is the first-in-class evidence that shape-changing drug delivery device below a millimeter, okay, so I'm not talking about above millimeter, can increase the efficiency of drug delivery in vivo. And this paper was featured on the November issue of the cover of Science Advances. Looking towards the future, we can also integrate chips on it. So I know a lot of people are interested in wearables. I'm very interested in wearables on the inside of the body. So if you are interested, I would love to talk to you. In this case, we have attached ID chip, so integrated electronics. Because of that, the device is a bit bigger because you can't buy very small RFID and LFID on the body. And we show in this case that we can tag a caterpillar. Okay, so this is a live with the chip. And we are hoping to see if we can have resident electronics inside the body in certain parts of the body. So just to summarize, this is a highly parallel wafer. This is just a 3-inch wafer. You can make many of them. We've demonstrated in vivo value for biopsy and drug delivery, and there's wafer scale integration. Looking towards the future, we are thinking of very highly programmed biochemical devices. one of the papers I won't have time to talk about, but please read this. This is, I think, one of the great paper. I was in Science in 2017, where with my collaborator Rebecca Schulman, we have developed a new kind of material based on DNA. And it's a DNA high swelling gel. And based on a DNA sequence, we can control which part of the device. Now, this is a gel-based device. It's not metal. But we can control which part of the device moves, opens or closes. Okay, so we have a material now, a digital kind of material based that can be actuated by a specific signal of DNA. So think about the macrophage again. We want a very specific biochemical to trigger the material. And this work, actually, the co-first author is in Seoul. He's did exceptional work on this paper. I won't have time, but we have a lot of materials with gels. They are reversible. They are biodegradable. I don't have time to talk about it. So, for example, they can close and they can also degrade. So one of the issues I told you in these devices, so a lot of people say my device is biocompatible. But biocompatible, it may not be enough, because if you put biocompatible things in your bloodstream, they can aggregate and you can get a stroke. So you may have to clear, just because it's biocompatible doesn't mean you can put it in the bloodstream, because they can aggregate and form a clot and block your vessel. So eventually, I think we all will need to think about whether we need not just biocompatible, but biodegradable. So we will need to work with materials that go away, because if you leave them in certain parts of the body, it might be fine. In the GI tract, in the brain, it might be fine. But if you're putting it in the bloodstream, there's always a risk of stroke or blocking the vessel. We do work on robotic control. I haven't had time to talk about it. This is a recent paper in Advanced Healthcare Materials on MR guidance of grippers. So we are thinking of guidance. But I find this very challenging to implement in vivo when I do a lot of this work, because as the size of the device goes very small, most of these methods will not work in vivo. So I'm still in favor of like autonomous devices that, like the macrophage, that signal, send some chemical and operate on their own. So with that, I'll end. If you're interested, we've written some review papers. This paper on basically arguing from that active matter can be used for tumors, to reach the interior of tumors, and also maybe clearing blood clots. We are very excited about this idea of hydrogel transformers. So I know I have a 9-year-old son. We always go to the store and we always get rigid transformers. Okay, you can get a tank, and you can make it into a ship. But I have never seen a soft transformer. Okay, so I would love if we could develop transformers that are made of gels, just like the human body, that switch shapes between different configurations. And I'm trying to do that with the DNA. So we are trying to switch the gel back and forth. We've done a lot of work with Sarthak Mishra on, using robotics and other things with soft gels. And, long ago, and I continue to work in this area, to try to integrate to make a sub-millimeter, like a pill cam, but with moving parts, with surgical parts, with shape change parts. With that, I'll end today, and I'll be happy to take questions. Thank you.
Yeah, thank you very much for a nice talk. So it's a Q&A time. So if you have any questions, just let me know. Yep.
Hi, thank you for an exciting talk. It's very fascinating. Great technology. I have a question about your inspiration. So your techniques were inspired by the macrophages. So I'm just wondering if you actually have tried to use a real macrophages molecular interaction on your devices, because they are really, really, the way that they detect the pathogen and they get activated is very, very precise. So I'm just wondering if instead of using like a thermal or enzymatic, have you actually used to try to like coat your gripper with macrophage molecules, for example, to make it really like a macrophage?
We are thinking about that. Unfortunately, these are very futuristic ideas. It's hard to get funding for them in the US especially, but that is a great idea we are thinking of. We are also thinking in our lab, we are currently working on technology to put tattoos on cells. So actually use macrophages, use cells, use bacteria, but also to put antibodies onto the grippers, and we are trying to do that. To my knowledge, I can't say that we have a very strong effort, but we have thought about that. That's a great idea. Yeah, I think either to use real cells and put, I like devices which, the problem with biopsy is that you need quite stiff materials to cut through the surface. If you use a macrophage, if you use the gel things, they are too soft. So they are good for clearing stuff in the bloodstream, etc. But if you want to do a tissue, they will not be able to get that. But yeah, that's a great idea. I think there's a lot of future for working with cells, and we already know these machines exist in the body, so yeah.
Okay, thank you. Just one more quick technical question. So you use a thermal responsive. So how sensitive is it? Because if you go into the body, your body is pretty homogeneous temperaturally, right? So how to control is opening and closing of the gripper.
So those grippers, they can only close. But the other gel grippers can open and close. They have a LCST. Many gels have a transition temperature. It's pretty precise. By synthetic chemistry, you can change the transition by putting different molecular weights of the gel. So you can have a gel that triggers around 37. So above 37 it will be open, below 37 it will close. Yeah.
Okay, thank you so much.
Okay, any questions? So I have a very quick question. So eventually you need to remove the grippers, micro grippers, right? So
no, not if they are biodegradable. So yeah,
okay, I see.
So we, John Rogers has pioneered transient electronics, but many of his ideas can be used for transient machines. So we have researched the literature, and there are thin films that dissolve in seconds, minutes, hours, days. So you can, by appropriate choice of film, you can have, like if you want something in the bloodstream, I think you need to have it disappear fairly rapidly. Otherwise, there's a risk of aggregation. If you aggregate particles in the bloodstream, you can block the flow of blood, and that wouldn't be good. So yeah, but in the GI tract, maybe days, things like that. Yeah.
Okay, yeah. Thank you very much for this nice talk. Yeah, thank you. So our next speaker is Professor Samuel Sanchez at Institute for Bioengineering of Catalonia. So the title is Swarms of Hybrid and Biocompatible Nanobots for Cancer Nanomedicine. Yeah, let's welcome Professor Sanchez.
Yeah, thanks for the kind invitation to come to this wonderful meeting. It's really nice, and thanks for making everything so easy to us, relatively easy, right, to fly all the way here. Yeah, so I'm also glad that our research has been named today a couple of times from the introduction, right, on the soft robotics, and then Joe talked about our catalytic, or now David about our nano drillers, or even Lizanne talk about the bacteria interaction. But I'm not going to talk about all this stuff today, so I'm going to focus on the things that we are doing lately in the last couple of years, which is what we call hybrid robotics or hybrid nanomotors. Is combining from both worlds, right? From artificial, we use nanoparticles, and from living systems, we use enzymes. All together will be something moving around, as you can see here. So we try to mimic what nature does, like bacteria and viruses, right? More or less the same size, make nanoparticles move, and then hopefully the swarming comes from the collective understanding and behavior of these nanobots. why cancer nanomedicine? So one of our ideas and the visions in this field is how to transfer things that have been done over the last couple of decades in nanomedicine all the way to nanorobotics, so give these nanoparticles activity, some active matter, and use it for cancer theranostics. Actually, I'm not going to talk about theranostics. I'm going to talk about more therapy or drug delivery systems. So still we are not doing anything in diagnostics, but that's one of the ideas. So I was quite sure something will happen. Actually, in that computer, everything was fine. So I don't know now with the phones. So check twice. Yeah, also to David, right? So anyway, nanoparticles have been done over the last, as I said, decades. But there's a recent metadata study which says that only 0.7 of the nanoparticles is designed to treat tumor risk to the target. So our proposition, our solution, is to do this nanorobotics, to take these nanoparticles and engage them with motion to give them self-propulsion and going at the nanoscale. So, but there are many challenges, many biological barriers that nanoparticles need to overcome in order to get to the target tissue. And these are some of the things that were reviewed in this nice paper by Chan and Poon. And we are also working on that. So at the organ level barrier, so you can find the protein corona formation. So people keep saying that we're going to put this nanoparticle in the bloodstream. So what happens with the protein that will be formed around that nanoparticles? So that's a challenge. So we're working on that. We have a paper in the archive that hopefully will be eventually published, but I will go into that if you want later on. Then once they go to suborgan level, so Per was talking about this matrix, this complexity, right, the media. So they go here, and then there's a very viscous media, very entangled tissues where they need to go through. So we hope the active nanoparticles will play an important role there. And then what happens when they get into the cell, there's a lot of issues there, right? So endosomal escape and so forth. So what we want to do is, instead of using passive nanoparticles to a cell and moving them, and this is one of the ideas, goes towards precision medicine, something that will go from point A to point B, but I can also drill into some tissues and go and penetrate. This is mimicking all the layers like a tumor can have, right, and penetrate inside. So that's a big challenge. So how we get particles moving? We have heard from Per today how to use catalysis to self-propel spherical particles. So what you have here is a catalytic material. In this case, we started using platinum. Platinum will be on one side, so it's always has been claimed you need this asymmetric reaction to happen, right? It will create, as it was said before, like a fluid flow around that, and you can, I mean, there have been many mechanisms proposed, like ionic or non-ionic diffuse phoresis. But anyway, you can do that from the micron scale all the way down to the nanoscale. Okay, and you have seen here how these nanoparticles move. So, but these are still, there's a big challenge to bring this into the nanomedicine world, right? So what I'm gonna ask to my newly designed nanoparticles are going through this chart. First, they need to be self-propelled, so you put them in solution. So you want to let them work autonomously, so not using any external field. Then you want the whole system to be biocompatible, okay? So it's not only the particle, but also the particle and the fluid and the fuel that will propel the particles, right? So as I just mentioned, like over the last years, we were using ourselves platinum. Platinum needs peroxide, which is toxic for the cells, right? So you need, the whole thing needs to be biocompatible. We are studying many fundamental aspects, and then drug delivery system targeting cancer cells, and in the future, or now, I will present some imaging in vivo. So which are the minimal components of this nanobot system? How do they look like? We again look into nature, and then we need to make something like, which is simple in a way, mimicking what we find like cancer, bacteria, and viruses. So you will see things that are the scale of tens of micron all the way to micron scale or hundreds of nanometers, and that's what we do in our lab. So that's the first component, to have a chassis, typical nanoparticle. Okay, so that's what we do here. And the only material I will be talking tomorrow I will talk about more materials in the other meeting, but today I will talk about mesoporous silica, which is a grass material. It's generally recognized as safe, so there's still some FDA-approved material or like application on this mesoporous silica, and now there's a recent study which shows that this is one of the promising materials in the inorganic field beyond the organic like liposomes we have here for biomedicine. So the second component needs to be a functional component, right? So we have seen platinum, magnetic materials this morning a lot. I will hear later. Titanium dioxide for photocatalytic, gold to use light to generate heat, cells even like to propel them. And what I will tell you today, how we use enzymes as biocatalytic functional component to move them. Why enzymes? Because they are bioavailable. You can find in your body like thousands of them, and for each enzyme there will be at least one substrate that can be catalyzed. So that opens many possibilities to find an application where there's an abundant substrate that will need an enzymes to propel and to generate a chemical gradient. Okay, we have seen also in literature how people use and claim different aspects and benefits from enzymes. A useman like a few years ago was explaining like chemotaxis of a cascade of enzymes where you have the substrate of an enzyme will lead to a product which will be in turn the substrate of the next one and so on. So you can propagate this signal over a long distance. People have claimed also chemotaxis on polymeric nanoparticle where they have enzyme on one side. And also we have seen the nice, beautiful work from Per Fischer where they used the urease enzymes to liquefy the mucus, but also you can use this cavity of the polymerisome, right, to have cascade of reactions. So there are many benefits of using enzymes in this reaction, and this account also that was part of this meeting, right, and a special issue a few years ago, like we summarized all the work from different people using different enzymes. But none of them were using one single type of particle and then systematically studied different enzymes. So we had selected these 4 that I mentioned here, right, which have different K-cut, and we studied the dynamics and the motion. So we use hollow microparticles, so mesoporosilica. Then we bound enzymes, we bound aldolase, glucose oxidase, acetylcholinesterase, and urease. We tracked the mean square displacement, and we saw that for some enzymes there's absolutely no motion. Then for some enzyme, acetylcholinesterase, we see slight increase, but the best enzyme was actually like urease. So there's a kind of correlation, non-linear, and we have all the molecular dynamic simulation following this in the second part of this paper on speed of the microparticles related to the K-cut, right? So from now on, the only enzyme you will see in my talk is urease, okay? So then I told you about this Janus particle, the asymmetric reaction, but we wanted to make something scalable, something simple. So we also they were asked today, like, can you make in bulk, right? So we wanted to see whether we can have a particle just put into enzyme solution and then see an asymmetric distribution as you can see here, right? So if you level the enzymes and you go to the confocal microscope, you will see this image, right? So in this image, you are not going to appreciate really clearly what's the enzymatic distribution, but if you use STORM, as you're seeing here, the blinking of every single label from the enzyme, you can resolve, and this is the ring around the particle, right, the enzymatic distribution, and you can also play with the 3D reconstruction, so every single dot is an enzyme here in 3D. But that, for me, was not enough. So what we did is to acquire like hundreds of these particles, right, in 3D and see the distribution as you will see in a second. But the point is that if you take urease and this is the ring again around the particle, you'll see patches, patchy-like structure. You will see gaps here. So that asymmetry is enough to create a fluid flow around the particle asymmetrically that will lead to this particle to motion. I will go back to that slide. Okay, so you take microparticles in this. This is the control. There's no urease, and this is the control with 100 millimolar of urea. Sorry, this was not urea, and this is with the fuel, and you can see cell-propel motion here. So the benefits are that you have enhanced transport, you have fluid mixing as well, so you have interaction with the cellular matrix, and you can reach the target faster. So I was telling you about the patchy-like structure, so you can compute, and then we have here an algorithm, right, to take all the particles and distribute asymmetrically, and the blue color will say that you have a high density, so in each particular case, you cannot predict a priori what will be the distribution, but you have always patchy-like will lead to motion. Okay, we have now very simple particle, mesoporosilica, you functionalize with enzyme, you put in solution. There's no metal there, there's no layer deposition, you don't need any expensive equipment. And you can also go and quantify the nanoscale. So these are nanoparticles, like a couple of 100 nanometers. This, every single dot is an enzyme. So this is how we improve the algorithm to get all the way here. Okay, this is from the raw data all the filters for months and months that now my students do much faster. I have no idea about this Python code and all the machine learning, right? But people have managed to get the distribution of enzymes, but not only that, so in this paper we also talk about the protein corona formation, so you can distinguish between the different proteins around. Okay, so this can be done for any type of particle. And here the only main point is not to show the versatility on our synthesis, but also the change in dynamics. So we're talking also today about the Brownian, how the Brownian fluctuation affects, right? For the nanoscale, I mean, the motion is almost negligible, so it's light, enhanced Brownian motion, but you go on to larger scale, so then you becomes more ballistic. So what's next? What can we do with them? So we have this pattern where we just have different components. So we have a particle that has an enzyme that can be for propulsion, but you can have a third component that can be like, we have 1.5 pages of this third component and all the plethora of application. But here I will tell you only about the transport. So how can we compare with passive particles? Demonstrated that having nanoparticles loaded with anti-cancer cell can be efficient drug delivery system. They can also be self-sensing using DNA nanoswitches together. And we did this from Francesco Ricci. DNA nanoswitches can detect the pH. Also, people from Valencia, Ramon Martinez, we have molecular gates to have trigger release. And I will tell you now about this 3D bladder cancer. So there are many uses that you can have with this mesoporous silica. And the way it works, you make the pores here, you can load the drugs inside. Okay, you can have the enzyme binding around. And then there are two types of delivery. One will be sustained release. So while it's moving, you have the drug being released without any control, but slowly. And then you can have the trigger release. So the sustained release will be as following. So you have the mesoporous silica again. You have then loaded with doxorubicin, this anti-cancer drug. So there's a tracking trajectory, the release profile, and this is HeLa cell. So every single dot here, red dot, will be a nanoparticle with a dox inside. If you have them loaded with urease, and then you have urea, then it will be self-propelled, right? But not only that, but the release profile will be much higher. I mean, you can quantify as well inside the fluorescence, and then we have seen an increase of release upon the amount of urea, and we think that there's a chemical reaction changing the it changes the pH, right? As per explained in the morning, so you go to pH 9, then you change the charges and the interaction with the drug, and the drug will be released much faster. So what you want to do is to kill the cells, of course. So then without urea, you see some of them inside after 6 hours. With a little bit of urea, you have many more inside, but not only that, viability decreases, but also like once they start to move around 25 to 50 millimolar, there's a huge difference in viability. Okay, just a few words about this intracellular release. So we have these molecular gates that open only when the pH is around 5.5. And then what we discover, I mean, the finding here is like you put with HeLa cell, they will go inside, and then we have the control with non-cancer cell, and then there will be no opening of the gate, so there's no release, and then there's no killing of the cells. Okay, and we have checked, I mean, this is the release of the drug at pH 5, but you can also check by different methods, by confocal, by flow cytometry. We see high content of fluorescence inside the cells. Okay, some little more pictures on where do they stay. And now we'll go back to the final application. Oh, how the time is not running, but it's fine, right? Okay, so what do we want to do with this? One of the main applications is to localize them and to study bladder cancer. So I told you about we have particles with urea. So urea is in our urine is in the bladder, and this is 9th most common cancer worldwide. But it is not the main important point. The main important is that if we happen to have bladder cancer, unfortunately, like 70% of us will go back to the hospital, and actually not for once, but for many years. So I know many people who are going continuously over 5 to 10 years to a hospital, right? So this high recurrence rate makes this most expensive cancer to treat worldwide. So we are focused on that, and the first thing we did is to replicate what happens in the bladder. So this kind of 3D structure. So we had the spheroid based on bladder cancer. We took the nanoparticle urease, and then we anchored also antibody, which is specifically targeting the bladder cancer, but not only that, it's also therapeutic. So you don't need to put any drug on the particle. Okay, so then we started to work on bladder cancer spheroids. We did it, many of them. So you have to have an antigen to see which are, marker to see the antigens that are overexpressed on the membrane, so you can bind with the antibody. So what we discover, I mean, in this case, we have hundreds of cells here making the spheroid. So if we add again the fuel, so you'll see some red level, red mark inside. So this is actually the case here. So we also put PEG to avoid, absorption and so on. So you will see an increase of delivery inside, but not only that, we've seen reduction on the tumor progression, about 40%, only having the nanobots with the urea inside. Okay, so just to summarize, what we have found so far is that we have gone in vitro from 2D to 3D culture. So we have enhanced delivery because we have a particle that moves around, and then we can target and penetrate into 3D tissue. Okay, so now what have we done in vivo? So yeah, in order to go to in vivo, we cannot talk about few particles. That makes no sense, right? So there are many studies that you need trillions of particles to treat tumor. So you need to start understanding collective behavior and have to have them moving around. And we have seen swarms today from Lizang, right? Like moving from one place to another. So ours are chemical propelled, not magnetic. And this is part of my ERC for the next upcoming year. So what I'm going to show you is a little bit of where are we heading up, right? So yeah, that's what you are seeing here is a video in real time. Like these are millions of optical video of millions of these nanoparticles moving in solution. So in a way, it's similar to what Per was talking today, right? About these convective flows, and then so we don't understand. We want to work on more on the fundamentals, but you know it's moving in 3D, so and these are nanoparticles, so complicated to analyze and understand anything in this sense. But of course, they are moving collectively. They're making these waves. So one of the interesting things here is that they are mixing the fluid around. Okay, so if you put them into the bladder, they will start mixing whatever is there. Okay, so that will have many benefits. So in order to see them, what we did together with collaborators from Cipio Magún in Spain is to generate the radio isotope or nanobot radio labeling, like fluorine or iodine. So we functionalize the particle. We also decorate it with gold, so we can put the iode here, iodine, and we clean them up, we separate, we characterize, and then now we have the nanoparticles labeled. So going back to the swarming, so this is about optical image. So if you add a droplet of millions of these nanoparticles without urea, they will just sediment and spread around. They are not going to move anywhere, as you can see here. Whereas if you have urea, they will start making these flows around. Okay, and you will see now a bit more, and not only the flows, but I mean the particles moving, but we made the particle image velocimetry, so we can also quantify and demonstrate that they move the fluid around. And then we did the same in vitro by PET-CT. So that was like, for us, a great experiment. So we have microchannels where we challenge like different paths of the motion of the particles with fluorine or iodine. Then here on the left side, you see the control, and then with urea on the right side. Now I will come and zoom in. So if you have a straight channel, there's not a big difference between the passive and the active. And these are swarms, a million of particles moving. But then if you start making meanders on different very sharp edges, then it will be more difficult for passive particles to move through, whereas for active particles, they will go to the other side of the channel much faster. Okay, this is still in vitro. So the next step was to bring them in vivo. So we had this systemic, like through the tail. The only point of this experiment was to demonstrate that this was not killing the mice. So mice were alive, that was fine. And then also to move forward, we wanted to just select one of the radio labels. So we selected fluorine, which was much more stable. You can see here iodine goes to different areas where we don't like them to go. So there is not very good absorption. Just to clarify, fluorine is chemically bound to the enzyme, so we don't need gold, we don't need any metal for tracking these nanobots, which is also beneficial. So the last experiment is the following. So I told you that we have this swarming moving around, it's moving the fluid. So one of the main problems in bladder cancer, that's why the recurrency comes, is that you have the patient laying down for half an hour, then the immunotherapy or the chemotherapy, the sediments. Okay, you empty the bladder, you fill the bladder with the drug, sediments, and then you need to rotate again every half an hour, and so on for 2 hours, right? So what we have here is something, if you have something that is moving around the bladder, so right, you can speed up the process, right? You don't need to move the patient around. So in this case, what we did, we empty the bladder of the mice, we inject intravesically the nanobots, and then we've seen that here, I mean, the red cloud where you're seeing is the nanobots touching the whole volume, okay? So it's reaching the whole lumen on the walls of the bladder where the cancer are, so this is main benefit. And for the other controls, you can see here the CT. This is always sedimenting, okay? Either you have with BSA, which is a model protein like non-active, of course, or you have the control without urea and so on. So all the controls will sediment, and this is very much less efficient than having the cloud of nanobots moving. Okay, so you can make all type of images, like different cuts from the bladder, different cases. So you can see time 0 is fully covering all the bladder, 45 minutes still remains there covering, but any other control, as you can see over time, again, is sedimenting, right, and not being efficient method. So we hope that, I mean, we have the interest from clinicians. We're working with patient-derived cells. We're injecting these in mice, and just, well, this is some quantification. I can just skip. And I can go directly to the future direction. We're working together with the hospital clinic in Barcelona and the other partners in this direction. So we managed to get the ethics, right, to generate the mouse model. So we injected bladder cancer cells. So we generated the model here. We're doing exactly the same clinical administration protocol. And you will see now, like, this is a summary of the slides. And you will see how we create the tumor, the mouse still alive for 15 days when we inject the nanobots. And the interesting point here, the main results are right pointed here. So you have urea and urease nanobots. They will actually accumulate. Let me just go to here. Yeah, much better, right? So here what you are seeing is the bladder tumor, okay? And this is the PET-CT of the nanobots accumulating the tumor. So for actually, the most exciting results come from the negative results, where we had nanoparticles there and they were not accumulated, and BSA control also, there's low mole accumulation and a huge increase when you have the nanoparticles accumulated here. So now we are moving to the therapeutics, so we are collaboration with the different companies who are using the drug in order to treat the bladder cancer tumor. So I hope, I don't know how fast I went, but probably too fast right now after lunch, but I hope I could convince you that we have chemically powered and self-powered nanobots that are autonomously moving. So this system is nowadays biocompatible, right? Where, as I said, moving in different materials in the future. We're also willing to study some fundamental aspects, active drug delivery, and all the way to in vivo. So this is one of the applications. So my plan was to talk about different applications probably in the next meeting, right? There are different ideas, but I think the most important is not only the motion, but the interaction with the extracellular matrix, with the organs, right, with the tissues. So that is what the enzymes, in this particular case, the urease is a great benefit. Okay, so other materials we use in MOFs, liposomes, PLGA, extracellular vesicles, polymeric magnetosomes, coarcivate. So just once having this idea of having a hybrid system, I think it opened to the interest of all these partners to collaborate with us and work on these nanoparticles. So one of the ideas is to go, and the question here, can we use these propellant nanoparticles to increase? I mean, we are not going to cure the tumor, I can tell you, right? Like I was saying many years ago, right? But yeah, can we increase the efficiency all the way? I don't know. Why not to dream 20% instead of 0.7%? So I have to thank, of course, all my team members, and in particular Tania and Ana Hortelao, who have just this from her graduation PhD a month ago. Mainly, so we have about 20 people in the group, but this work that I presented today is mainly from they both, and also a bit from Xavi Arqué, the funding and the collaborations, and thanks for your attention.
Yeah. Thank you. Thank you very much for this nice talk. Yeah. Questions, please.
I have one probably quick question. I think perhaps I missed some of the point that you addressed, but so the bladder cancer targeted delivery, you have a major for silica, and do you have an antibody that will direct into the
anti-FGFR
okay,
FR3.
And then at the same time you have a urease, right?
Yes.
So the enzyme that propels.
Exactly.
Yeah. And then you said that one of the last slide was phenomenal because you said 20% of targeting efficiency. Then how do you measure those? I mean, you have a measure for structure, you have an enzyme, you have a antibody that are targeted into the tumor and so on. So it's a very kind of maybe perhaps a synergistic effect. But how do you quantify each component that will drop into some numbers?
That's a very good question. Doing all the controls, right?
Yeah.
So, but in vivo, in vivo, the challenge, right, is you need to sacrifice animals to do these controls, and we had we are doing this, and there's some interesting and also some disappointing results in that direction. So we have, the way you do is you take the passive nanoparticles, you inject them, right? Particles with the radio label, particles with enzymes, particles with enzymes and antibody, particles with antibodies only, right? So, having the highest has been nanoparticles without antibody. And that's 20% higher than nanoparticles with enzyme, that's the best. Okay. Nanoparticles with it is 20% higher than nanoparticles with antibody, but still it's a huge difference compared with all the other controls.
Right.
So of course antibody is really good for targeting.
But this is not the systemic injection. It's not.
This is intravesical injection, definitely.
Right.
Yes.
Right.
Yes.
And then so you have a urease, and then you generate. Is it Janus? Anisotropic nanoparticle, or is that just you just call it homogeneous?
No, there's not homogeneous. That's why in the first part of the talk I was talking about this super resolution microscopy to demonstrate to convince you that we have a certain asymmetry, molecular asymmetry.
I think that's the point.
Yes.
Right.
Okay. Otherwise you won't get any motion.
Yeah. I think this is great. I mean, I'm really looking forward to the next results coming up probably soon, and this is. And related question about the enzyme propelled system. So is the amine generation, or how do you really propel it? I mean, is that Parrot mentioned about that? Is that related thing or not? The proportion. I mean, is that?
So the fact that we have the chemical reaction producing ammonia and CO2, right? Generating a chemical gradient around the particle that generates at the same time a fluid flow.
Okay.
Right. Okay. An asymmetric fluid flow, and then this gives a vector velocity of a particle.
But it goes, anywhere, right? It's just goes almost everywhere, right? It's not directed.
No, it's not directed because it's still at the nanoscale, right? Okay. So the rotational diffusion is very high, so it keeps rotating. Yes. Okay. But that's why I think the interesting point here when we talk about nanomedicine is the collective phenomenon, right? So that's why I know I went very fast, right? But in that the main point of that slide with nanoparticles, microparticles, and microtubes was that the microparticles is directional. Okay. Right? And the larger they are, they keep more straight all the time. Okay. The nanoparticles will just have enhanced Brownian motion. But you put them all together, you generate this collective fluid flow.
Yeah, and
I think Per wants to add something. Per wants to add something.
Yeah, thanks. Yeah, I guess there's this question about does the individual one move or not. But I think from your injection and your particle imaging velocimetry, it's also very interesting that I think you see probably the way it's injected that the particles are all together initially in one location, and their activity will then create a huge concentration difference, which will cause enormous convection flows, which means they all get washed along. So it doesn't really matter all that much if the individual one is all that active. So I think an interesting experiment would be if you were to take particles and coat them so they're more symmetric, or take slightly larger ones so that your random asymmetry drops out. And probably, I would guess, by injecting and convective flow, you nevertheless achieve nice distribution.
So So you mean if they're even if they are fully coated with enzymes, they will
Yes,
still
I'm
generate
wondering
that.
the fact that you basically have a region where there's a lot of chemical activity, because in your PIV, the convective flows are just so much bigger than the individual motion.
Absolutely.
It could simplify the design and yet be very effective. And I'm guessing if you were to do it symmetric, it would still work.
Yeah, well, I don't know. That's a very good point. So collectively, they will generate the flow, and the fact that they are symmetrically coated, I don't know if it's easier than what we do, to be honest, right? So, so actually, like we struggle. We try to do systematically from the nano.
If the particle is larger, then your relative difference is less, and then then statistical fluctuations will do less. So just taking larger ones might do it.
Yeah. So we struggle to get the same particle from the 100 nano to the micro particle size. But yeah, you can buy them. Like yes.
I have a more general questions. So you injected those kind of enzymatic particle into the bladder system. I think because in the bladder the flow is very low. But then I'm wondering if those kind of technology can be used for the bloodstream, and then how fast it should move, and then then how much force is required to be used in those kind of flow system.
Yeah. So, I was very happy to focus on this niche, right, on the bladder cancer, because probably if I have given this talk 3 years ago, probably last time I came to Korea, I was talking about fantastic voyage and put in the bloodstream, which to me right now, we cannot compete with that. So we're talking, I mean, the long range is dominated by the flow of the blood, right? So only when you find spots, places where there's a high viscosity, low diffusion, or there's not even like nutrients getting there, is when we can even think about that this makes sense, right? So I don't think I can put down the numbers. So we have a couple of papers to people from Tübingen measuring the force goes from femtonewton, 60 femtonewton, the nanoparticles, to piconewton, right? 16 piconewton, the microparticles. So I mean, this is nothing, right? Yeah. So I'm thinking on these particular places. I think there are many questions here from the chat.
So, yeah. So we have an online question. So some people asked about the speed of the your particle. So the last question is, the speed will be lower in the case of the non-ionized particle. So what's your opinion on that? Yeah.
Or just asymmetric. So I'm really thinking speed of urease nanoparticles made by pure Janus particles, different from nanoparticles, asymmetric. Oh yeah, okay. So whether having a Janus particle or asymmetrically with enzymes will, as I started to say before, like we struggle to get exactly, to make a systematic study, you have to have the same type of particle, right? Half coated and non-half coated. So our initial attempts is that the Janus particle gives higher speed than the patchy-like extraction that we have. But we haven't published that because at the end is what we were discussing, right? We stopped caring about single particle velocity. At the end is the collective phenomena. But this is a good point, and indeed the listener is right.
Okay, so let's give a big hand to Professor Sanchez. Yeah. So actually, we were supposed to have to skip the coffee break, but by considering the jet lag of speakers, so probably it's better to have the coffee break. So we will start at 3:20. Okay. Yeah. Thank you. Yeah.
So when we convene?
Yeah. So we will have the coffee break, and then we can start at 3:20. Okay. Yeah.
Thank you. Okay, everyone, we are just about to start the final session of today's conference, so please get back to seat, please. And yes, the next presenter would be Professor Nelson.
Sure, I'll put it here. So. Is this working now, or are we all okay?
Yeah, I'm gonna. Okay, it's work. Okay, so my name is Woo-Young Shim at the Yonsei University at the Material Science Department. It's very honor and my pleasure to join in this great session. Well, in fact, I was born and raised in the Seoul area, and I've never seen this great, fantastic view in this office right here. So it's great. Anyway, so especially this is great. I think it's 19 years ago when we have a World Cup in Korea. So it's called the Red Devil. It's a supporter, soccer supporter. Get around in all this area, and I still remember those fantastic days. Especially many speakers from Europe. So I mentioned soccer. Okay. So the session of today and our first talk is from Professor Bradley Nelson. Professor Nelson is a professor of robotics and intelligent system at ETH, and he's also a department head of mechanical and process engineering at the same institute. He had over 35 years of experience in the field of robotics and received many numbers of awards in the related field. And he also advisory board and editorial board in many renowned international journals. He's gonna give us talk about micro robotics and nanomedicine. Let's welcome Professor Nelson.
Okay, thank you very much. Thank you to the organizers for a wonderful organization. It's always great to be back in Korea, and it is a lovely view, isn't it? It is nice up here. So I'm going to talk today about microrobotics, nanomedicine. You know, we've been thinking about how to make small, intelligent devices for about 18 years now is when we started actively doing this. I'm a mechanical engineer, so I'm coming at this maybe more from a device perspective, and I think the longer I get into it, the more important I think it is that we think about it from the device perspective, so both from the top down and the bottom up. I think we want to have both of those perspectives, and so you'll see that here. I've had a great group over the years that have come through my lab. Many of these people are still with me. Some of them are off doing bigger and better things. But it's we usually keep around 40, 45 people in the group, and we have a lot of different skill sets. Like I said, I'm a mechanical engineer, a roboticist, and then Salvador Pane, he's my co-director, a professor at ETH as well. He works very closely with me. He's a chemist, so he does all the material stuff and all the kinds of things that I've been hearing about this morning a lot. But Coming at this from more of from the medical device industry, I think it's, we all know the incredible success we've seen in the medical robotics field and all of the money that's being made, resources being invested, companies being acquired. But I think there's a couple of more recent developments that I think are positive for us on the small side as well, and that is Auris Health and their acquisition by Johnson & Johnson, as well as Intuitive Surgical's new ion system, which is coming to market. And both of these are basically robotically guided small catheters or small endoscopes, still relatively large by our standards, but 3 to 4 millimeters in size, that go deeper and deeper, in this case into the lungs, to get biopsies and to be able to do an analysis that you really can't do everything with CT and with MR. And so I think what that shows to me, or what I see in that trend is that we're kind of coming together where I'm a firm believer a lot of our devices, we're going to have to think about delivery systems, and we're going to have to think about how we bring those closely together. So this is where we are on the micro nano robotics side, and then the medical device here, they simply can't get much smaller. And so we're going to so it's natural then, as we're trying to treat bladder cancer, as we were hearing, or something in the kidneys, or in the heart, or in the brain, atherosclerosis, these kinds of things, that we're going to be able to want to go to places that we simply can't get with regular catheter approaches. And so when we think about how we do this through the surgical through the surgical workflow and in the operating room, you can see how these kinds of technologies are gonna merge and should be merging. But along the way, when we first started working on micro robots back in 2003 was when we first started thinking about how we were going to use magnetic fields to do this. Just go back to the basic physics and remind ourselves that we generate the torques with fields, and we generate the forces with the field gradients. And this is the point Pierre made earlier, is that it's hard to push things into the body. It's easier to pull them out because of this. And so these physics are important. One of the things as a mechanical engineer, as a roboticist, that we brought to the field was how we could analyze this, look at this from a robotics perspective. And so kind of my fun theoretical work these days, we're working on a few papers that are going to be coming out on how we really can look at this interaction of magnetic fields, field gradients, electromagnetic coils, and really put that in the exact same kind of framework that robot manipulators, which are ubiquitous, right? There's millions of these all over the world. The whole mathematics of it, the whole definition of that's really what the contribution of the robotics field has been. And so we're looking at all these fields that are being generated that we've heard about today, and trying to place that within that framework. And it's quite an interesting task to see the similarities and differences there. But I'm not going to spend time on that today. This is something that probably in another year or so I'll start talking a little bit more about. But one thing we have done over the years, spent a lot of time building electromagnetic navigation systems. And one of our insights, again going back to the robotics field, was the way you can precisely control small bodies. You can control their orientation and their position by generating the forces and torques appropriately. And so this is our first system called the Octomag. It has 8 electromagnetic coils for good reason. There's good mathematical reasons you want 8. It's the minimum you need in order to move a device with 5 degrees of freedom. That's something nobody ever knew before we published that paper in 2015. I entered the field thinking if I want 5 degrees of freedom, I want 5 separate actuators, and it doesn't work that way. It's a little more complicated. We've gone smaller, so you can buy these desktop systems, marketed those as, we used to call them Nanomag or Mini-Mag. Now I think they have another commercial name they're used. We also went bigger, small, with large animal systems, thinking, okay, if we're really going to do clinical work, we've got to do it at human scales. We can't do everything under a microscope and say, oh, it extends to big scales. And we've even brought systems to the clinic. And then some of those lessons we started going back smaller, and that's where we are right now, are with some of these small portable systems, trying to generate the fields and gradients that are going to be relevant. We have gone and done a lot of work on micro robots and moving them in vivo. moving them in animals, in rabbits, in rats, mice, and but I'm going to start first talking more about some of the, well, this is some of the rabbit work we did initially with 300 micron diameter devices that we can maneuver and puncture veins on the retina, things like that for eye surgery. But I want to talk more about what start off by coming at it from the device, from the top level down, and that is where I take my microrobot, I put it on a tether. In other words, I take a catheter and I put a magnet on the end of it, and I can use the same mathematics then that I use for guiding my microrobots with a few little tweaks to move catheters very dexterously in the body. This is an example of a cardiac ablation procedure in the heart to treat heart arrhythmias. We've learned a lot along the way on how you bring a product to an idea from the lab to the clinic, the need for investment, the need for following medical device regulations, and the need for working within surgical workflow as well as the hospital purchasing rules. This is our AN Focus, which we came out with around 5-6 years ago. You'll notice how it's a part here. It's an electromagnet, so I can turn it off. The fields are off. It's a regular operating room. You can prep the patient normally, operate on them like this, or you bring it together, turn the electromagnets on, and then you can guide the catheter with the patient inside. And here's where the surgeon's going to sit while doing the surgery. We've learned a lot over the years with this, treating patients. This is our electrophysiologist here doing an ablation procedure on a woman who's by herself, awake during the procedure. He's planning in her right atrium where he wants to ablate. And then he's guiding the catheter with this joystick. He likes to say he can drink coffee and do surgery at the same time, although I've never seen him actually do it, but he says he can. So the couple things we learned. I mean, the systems work very well. It's nice when you have an electromagnet with very few moving parts. It's really just the pump for cooling. These things, you turn them on, they work for a long time. They're very, very reliable. You got some training. There's a lot of surgeons are used to doing things. They train in certain ways. For instance, with the catheters, pushing, pulling, and turning, they have a real tactile feel for it. And then when you want to bring them back and have them use a joystick, that's a new thing. And so training is important for these kinds of things. But the other thing is, as we went through medical device regulations, we were using off-the-shelf catheters. They didn't work appropriately for some of the things we needed. And then also working with hospitals, realizing that large systems are difficult to get into hospitals, especially when it's new ideas that aren't normal. It's a challenge to convince a hospital to spend a couple $1 million and reserve a room just for your device. And so we went down this path and started looking at a couple things. One was the catheter problem. The catheters are the things that are eventually going to be delivering our microrobots or nanorobots. And so we started rethinking the way catheters were made. One of the things we came up with, one of the patents that we actually have been granted, is a variable stiffness catheter where we can change the compliance of the catheter along different sections of it. We use both low melting point alloys. In this case, we also use thermoset polymers. So these are devices that you heat them just above the body temperature, and then they get much more flexible. And then we can use our magnetic fields to move them and put them into positions that you simply can't put any current catheters. And so we can make them smaller than 1 millimeter in diameter. So these are small, and I think at some point this was what we think is the world's smallest steerable catheter. I don't know if it's still true, but it was at some point. The other thing that we realized during this path was the magnetic systems themselves, and so. And how those work. I think this morning during Zhao's presentation, you saw I saw an old Stereotaxis permanent magnet system in there, and he was moving things with permanent magnets. This is a system that's in well over, I don't know, over 100 of these around. I know there's at least one in Korea. This is the new Genesis system. The old they require an entire operating room has to be remodeled fixed up. So you've taken some of, and they can't be used for other things because you've got these large permanent magnets that are always got a magnetic field. They also have to be located quite a ways from any magnetic resonance imaging machines because the fields are strong enough that they can interfere with that. Magnetex is another company that made, and I believe there was one at one point here in Seoul, but I don't believe it's here anymore. This is a company out of Santa Monica that tried electromagnets as well. But both of these systems require dedicated rooms, and I mentioned the advantage of our hybrid system, but what we realized would be a much easier way to get into the clinic is to go with something that's portable, and so that's where we are right now with this Navion. We've gone from 7 tons to about 350 kilograms. Depending on how you define the workspace and where you want to put it, you can generate up to 40 millitesla or so with it, or you can put 2 of them together and create even larger fields. But they can be wheeled in and out of an operating room quite easily, set up in less than 10 minutes. They would just require some water and power to get them to move. And so that opened up a lot of possibilities for us. And so we're in the process now of going through regulatory procedures to get this approved to be used for clinical use. We continue to work on magnetic catheters and endoscopes and doing a lot of design in that. A key component of this is the advancer that pushes the device. Remember, a catheter endoscope, basically you push, you pull, and you turn it. This just pushes and pulls, and then the bending is taken care of by the magnetic fields. So that's something that we're currently in the process of trying to get out into hospitals and continuing down that path with it. Here's an example in my lab in CLA. Here's a fluoroscope. This is in the building I'm in at ETH, I should say. Controlled by a PlayStation controller. Horus Health looks more like an Xbox. We're using, and this represents an aortic arch trying to maneuver up into the carotid arteries, getting up deeper into the brain. This is at human scales. This is the vasculature. This is from actually a silicon model of a vasculature we got from a company called Trandomed in China. This human scale, and shows that our device works at human scales, and it allows, if you work with neurovascular surgeons, you saw this morning, there's a lot of different catheters they use. There's a lot of tricks they use to get those up there, and this is basically one catheter can do quite a bit here. We also can use this with our variable stiffness material, where we can put the catheter up in the aortic arch, generate enough field to bend it to get into the aorta, and then we can stiffen it and allow it to sit there to provide more stability then to a microcatheter that can come out and move even further up, deeper into the brain. And remember, the Navion is sitting up here, our magnetic system, so it's actually getting stronger and stronger fields the closer we get, so we're getting stronger dexterity on it. And then one of the other innovations in catheters, we have something we call a torque catheter. My marketing, I should say, the CEO of our new company, he likes to call it a walking catheter, but it's a device that kind of telescopes in a way. Again, patent filed on that, and is getting, but I don't think you'll find a catheter as dexterous as this one that can move, can literally tie itself in knots if you wanted to do that with it. So that gives us some ways to get into locations that are deeper, where then we can release the devices that we want to. This is a neurovascular surgeon we work with. This is a pig under here. He's standing next to it, but he could be 1,000 miles away with his Xbox, with the PlayStation controller here. And this is, if you look, this is a 2 French or 700 micron diameter catheter that's coming all the way up here through all of these twists and turns. And going up deeper and deeper into the neurovascular region of this pig that's alive at the time. And here's another same pig where he's pulled it out now, and he wants to go down now instead this route. And this is all with the exact same catheter. He hasn't changed catheters, and so there's no catheters out there, right? No neurovascular surgeons that could do that. But I think you'll see him, coming. Right there, making this sharp turn here. I mean, it doesn't look like a big deal to you, but when he did it, he says, I couldn't do this. I wouldn't be able to do this any other way. So I think that's allowing us now to get into locations, but we're still limited. And if you talk to neurovascular surgeons after patients suffer from a stroke and they do a thrombectomy and pull it out, they still think, patients still suffer. They're not getting everything. And so that's where I think the microrobot stuff comes in, right? Where we can actually go into some of these smaller vessels in the brain and dissolve some of these clots. So I think there's, it's getting close, and then releasing the microrobots in a way. And then the last part of the talk here, I want to just spend on some of our microrobots. We've spent several years on our similar to Pierre's group on our inspiration from bacterial flagella. We rotate magnetic fields. There's a lot of good physical reasons for this. The drag, rotational drag scales with a cube of length, so that helps us. And of course, we're generating torques here, not just forces. We're not worried about gradients, and so that also helps us. So there's a lot of physics, simple physics, that is on our side here. And then as we've gone down this over the years, new fabrication methods have become available, commercially available, like direct laser writing, which lets us make more and more complicated structures. Also, the other big part of the work has been on functionalizing these, putting 80,000 of them with a near-infrared dye tagged on them into a living mouse. With that near-infrared dye, we can actually see it in the peritoneal cavity. We can see the fluorescence through the skin and track the functionalized ABF swarm inside. Again, that's 80,000 of these. This is kind of what it looks like. The image you get, and what it would, this is in a petri dish, but what we imagine is going on, if we can see individual ones inside of the living mouse, is done quite a few years ago, though. All these devices are pretty rigid, and so we started thinking several years ago about adaptive materials, and also became concerned that as we entered this field, those first ABFs I showed you were made out of things like silicon germanium, gallium arsenide, nasty semiconductor kind of stuff. Then we were able to move to more biocompatible polymers, and now even biodegradable devices that are photocurable. But we're also interested in morphologies and looking at microorganisms that change their morphology and their life cycle. For instance, this parasite Trypanosoma brucei, which has a motile form and a stubby form, or a long slender form where it can swim, and a more motile form. And so we started thinking about how we might use that, be inspired by that, and then create devices. And again, all the materials, we started focusing more on biocompatible materials in this case. So polymers that we felt like, would have a chance of getting through regulatory pathways, iron oxide. We do have some graphene flakes in here to absorb radiation, so that when we heat it, it absorbs a lot of that radiation and then causes a shape change in one of the bilayers from a temperature change. And so we can go between this slender form and this motile form. And so this is an interesting process, because it allowed us to try a lot of different designs and go through, one of the joys of working at low Reynolds number flow is that everything's kind of analytical, and you can actually do some interesting models and kind of predict performance and then verify them. But they're still not quite, you've got an initial design, and we haven't really moved. David was talking about autonomy and trying to make things more autonomous. I fully believe that's the way to go. But we'd also like to make them programmable, and so we started working with some folks at Laura Heiderman's group, who was into nanomagnets. And we'd learned some interesting things. I guess I kind of knew, but I hadn't really thought about what the possibilities were. And we're all familiar with superparamagnetic single domain nanoparticles, and then of course multi-domain devices. But there's a region in here where you get a single domain, and the shape of if you can control the shape, you can actually have a big impact with the same material on the coercivity of the device itself. And so by patterning the same material, in this case cobalt platinum, with different scales, I can generate. My anisotropy curves didn't come out. I have a whole bunch of anisotropy curves that disappeared here, basically showing different coercivities on all of these. So these were the smaller, the lower the coercivity, the larger, the higher the coercivity. Yeah, some of these, something's happened to all my videos are messed up here. Oh, that's too bad. Oh well. So let me real quickly go through this. I've only got a few minutes anyway. Basically, what we do is we can take soft structures, which are bendable here, and if we put with the same material, just pattern different size nanomagnets on them, I can encode the device so that I can change the magnetization of each of these. Remember, they have a significant coercivity, so you can consider them hard magnets. And so putting, if I put this 4-square pattern here and I put a strong field there, okay, these guys aren't going to do anything. These two panels are going to align, but then I put a little bit of a weaker one. This guy will flip his domain, but flip his magnetization, but he won't, and then I can do it in the same way. So I've got with these 4 panels, I can program it to have 16 different kinds of magnetization, and that means it's going to exhibit 16 different behaviors to the same magnetic field. And so what the idea here is to create a kind of a programmable device with this concept of using nanomagnets and how the coercivity of these devices changes with size. And so I can, some nice little videos that didn't come out here, but anyway, these are the panels. This is the magnetization, but I was kind of explaining to you. And then my Tianyuan Huang, he's at Peking University now. He actually created a micro origami crane here that so that we could actually take this little tiny bird that's on the order of 20 microns or so, if I remember right, in length, and by just reprogramming it, we can get it to sort of fly, hover, and exhibit all these different kinds of bird-like behaviors, which let's see. So these are the different panels on there. Let me see if I can. Okay. Oh, these are showing. Okay. So then, no, that's not. Let me. Yeah. Okay. So anyway, there were videos showing each of these, but anyway, you get the idea. These are showing the way they're going to move. So. But anyway, so there's one idea of how you can, we can reprogram, we can re-encode these as we show. So just to summarize, electromagnetism, we're still, interesting work I know we saw with Joe Wong and some of the other folks here on chemically driven microrobots. I'm still excited about magnetism. One of the reasons is because I think it works with large devices better than with small devices, but we can bring them all together, I think, and once we get comfortable of getting our electromagnetic systems in operating rooms for guiding endoscopes and catheters, we can use the same system for guiding microrobots. We can use nanomagnetism. I think that's an interesting concept of changing the sizes and changing coercivities with the same material so that you can re-encode these devices and get them to behave differently if you'd like. I think there's a lot more to explore experimentally and theoretically, and that's what we're continuing to do, while we're also trying to get this out, at least the first infrastructure, this into clinics and into hospitals for so that it starts working its way into the surgical workflow. So I think that's all I have, and I had 2 videos here that everything kind of got. I should have gone through the whole talk. I thought everything was working, it wasn't, but oh well. Sorry. Anyway, thanks. Darn, I'm done. I'm happy to answer questions. So.
Yeah. So before taking questions, maybe you, so someone who controls the slide, as you can get out the slides they show, maybe you can play the video inserted into the slide. Can you go out? So finish the slideshow and go back to individual slide.
Oh, yeah.
Maybe you can.
They're on there.
Yeah, maybe you can play video.
Just bring that up. Yeah, see if that works.
Yeah.
Oh, yeah.
Okay.
Huh.
Okay.
Yeah, so here, this is the 4 panel. This shows 4 of the different behaviors. The same magnetic field, just showing different. So same thing, just re-encoded, basically reprogrammed in a way. What I'm showing there. And let me see if I can go to the next. All these different panels on there, and then yeah, there's a flapping, kind of a turning, hovering, and a kind of side slipping. So again, the same bird, the same crane, but just reprogrammed to behave differently depending on what you're doing here. And then these are some of the these are the things that didn't show up there. So, okay, good,
great. So,
thank you.
Any questions from audience first?
Great talk. Then the systems are really impressive. I'm wondering if you have a number just to know if you're pushing this catheter. It probably has to do about a meter or so, probably by the time it reaches the brain. And then at the end it's 700 microns. What forces do you put at the end of this catheter to actually advance it with all the friction all the way up to the brain? Is there a number?
I don't know the number. No, nobody. I don't know if anybody knows that number. We have measured it. Let's see. I'm trying to think in one of our projects. But I couldn't tell you I can't tell you off the top of my head right now.
At the end, it's probably some kind of syringe drive or something like this. Is that right? To advance it, it's sort of a.
So yeah, I mean, normally it's just 2 wheels that push it with the advancer. Now the torque catheter actually is a tiny little threaded device that spins its way up, and so it's pushing itself as well as pulling itself through the vasculature.
Because I'm wondering what the compressive strains are on a system like this, or what.
Well, so you get these things called huckling. There's a term huckling. It's kind of like buckling. It's like a helix, and it will wind itself up. So there's actually some interesting mechanics underlying all this, and you have to So you have to keep your tension proper in it, and if you have too much open space at a certain location and you do it right, it can start to go into this huckling mode. But huckling is the term. But that's one of the tricks. I mean, so you've got to get these.
And just to finish off, there's probably then no point in actually pushing for the 100 micron endoscope, which is something you can do over 10 centimeters. But the point is, it doesn't make any sense because you have to maintain this structure over a meter or so.
Yeah, because usually, I mean, a lot of catheters are about 1.8 meters long where they start. They go in here and come up. But so one of the advantages that we think with these new approaches is that you could actually start doing a radial coming in from the radial artery in the arm and going up. And there's there's a couple advantages. First of all, there are complications from your femoral artery. Patients have to lay in bed longer for a day. If it's in the radial artery, it can it's less of an issue. You can imagine why you don't get the pressure on it from standing up. But the problem, no neurosurgeons do that because they can't make those bends. Some cardiovascular people going into the heart will do that approach. And so that would be one of the potential advantages, and especially if you're going to do this remotely, say as Zhao mentioned before, we can do remote surgery and all those things. You want to do procedures that are going to have fewer complications, and so that would be one of the benefits. But anyway, that's not exactly what you're asking. But if you had it, if you're going to go 100 microns, I mean, so what people are doing and what patents are out there are these what they call flow-driven catheters. Have you seen this? So they're on little threads, and the end of it is the micro-robot has a special shape that is actually being driven with the flow so that you can go deeper. And so instead of pushing it, it's being pulled with the flow. There's a few groups that have worked on that, and there's a few patents that have been filed recently on the idea. We've played with it. We've played with it.
Like a parachute.
It's like a parachute kind of thing, yeah.
Yeah, I have a small question about the magnetic origami nanostructure. I think microstructure actually.
Yeah.
So I think it's fascinating. I read that paper in Nature. It was fascinating, And then actually my research group is curious about, what is the direction that you wanna take from that, all the motion controlled, beautiful, bird, right? And then do you wanna make it smaller, and what would be the limitation that you have? Because it's, you delivered the concept, and then where do you wanna go from there?
We're continuing on it. I'm not sure I want to say everything we're doing yet, because I think we're going to file some IP on it. Usually we go small because that's interesting, and then we find out when we go bigger. Let's see, small is easier to publish and to find the really hard problems on, but as we go bigger, we start to find applications that we think. Okay. And so I think remagnetizing things it's not a new idea, but I think there's a lot that can be done for medical devices if you can remagnetize them in vivo that will allow them to behave differently.
So the David gave a beautiful work on the grippers, and then something like that. You can easily use that for delivery vehicle. In certain purpose, and then that's easily done, right? Yeah. And then you can, unfold, your cargo into targeted area or something like that.
Right.
It could be a drug, it could be a, cells, enzymes, whatever you want to take with you. So that's a beautiful structure.
But I think, if you can, change your magnetization vector, or change the relationships of different parts, then all of a sudden you can create different behaviors in some very interesting ways. But I got a PhD student working on that right now. So.
Okay. All right. Thank you.
Okay. So I'll take on one more last question. Okay.
Thank you very much. My question is regarding actually the patient, since this is already very far towards application. So nowadays more and more people like to get tattoos, and tattoo inks are often magnetic. So I was wondering, how does it interfere maybe with this magnetic actuation in general? And if so, for instance, they cannot take any MRIs if they have large tattoos. Is that also a limitation then to this patient?
I don't think this. So I think the issue with MRs is not the magnitude of the field. It's more the pulse that you're the signal you're driving knocking the spins out with. And so our variation, our frequencies are very low. So I don't anticipate an issue with that. But that's okay. I should have told my daughter that before you got her tattoo. So good point. I should have.
Maybe you can engineer the position of your tattoo in every single thing. Right. Okay. Well,
great talk as usual. I'm impressed. Just one of my students typed in the question on the YouTube, so I just delivered the question for him. So basically his question is, can you use the Navion for untethered microbot to control? And also he asked, what's the working space of the Navion?
Yeah, so the Navion only right now has 3 coils in it. If you put it in 2 facing, then you could do some things with it. For microrobots, that would be potentially interesting. The workspace, it depends, right? It depends on how much field you want. That's the difference with a robot. If a robot can reach something or not. With a magnetic system, it's a little more complicated, but it's on the order of 10 inches on 25 centimeters or so by 25 centimeters, about a cube is what we're looking for. We'll have to get closer enough there.
Okay, thank you. I think that should answer to my student.
Good.
Okay, so let's thank Professor Ness again. Thank you. Okay, so last talk of the day is from Professor Hong-Soo Chang. He's the professor and chair in the Department of Robotics and Engineering in the Daegu Gyeongbuk Institute of Science Technology. So Daegu Gyeongbuk means Daegu is a city and Gyeongbuk is province. So it's Daegu located in Gyeongbuk province. Well, it looks like everybody knows about it already. Okay. Also, he's director and a center of a targeted neural network reconstruction of the National Science Challenge Initiative, and a co-director of the DGIST and ETH Microrobotics Research Center. So he's going to give us talk about the micro robots for cell delivery and active connection. Let's welcome Professor Che.
Thank you for a nice introduction. And I really appreciate that I can join this wonderful event. And I thank all the organizers. It's a wonderful organization. And I also, before I start, let me thank Brad. As the chair said, I'm leading DCTH Microbe Research Center. That means I learned microbe from Brad. So my background is in MEMS. So before I joined DIGIS, I was working on MEMS and piezoelectric devices. And after I joined the DIGIS, which is 2010, I started to learn what is microbe. So I was not his student or his postdoc, but after I became faculty member, I started to work with him and learned what is microbot, what is nanorobot, and my career on this side started from there. So I appreciate Brad. So I think today many leading professors, they already introduced what is a microbot, what is a nanorobot. So I think I can skip basically. So I think this is also, I think I can skip, but the message is I'm also trying to develop microbot or nanorobot to be used for biomedical applications. So basically, I'm dreaming that I can make a device that can be used for the surgeon, and the surgeon, regular surgeon, became super surgeon. So they may have a fantastic tool to treat their patient. So just briefly, as you know, we can control the microbot in a precise way, in a remotely way too, and also depends on target area, depends on the disease. We may be able to optimize the structures of micro nanostructures. So depends on the application, we can develop different types of beta structures. So by combining better structures and precise control, probably we can apply those microbot or nanorobot for various targeted therapy, such as precise cell or drug delivery, brachytherapy, hypothermia, generating heat in a local area, or multiple cell or drug delivery. So today I'm going to talk mostly on cell delivery. I mean, we do have some drug delivery research, but I think today let me share the information on cell delivery. So by combining all three previously explained technologies, probably our general aim is a minimally or non-invasive treatment. And as Brad said, potentially we can have a telemetry treatment. And previously, other speakers, David, mentioned about precision medicine, and Samuel also mentioned about precision medicine. So I think this is one of the kind of direction to go for us. So in general, for precision medicine, we need to understand the patient. And so we bring the samples from the patient and do sequencing, and we understand what the problem is. And also we make some organoid from the patient using the samples from the patient and mix different types of drugs and find out what's the best combination of the drug for this patient. And we also can make a xenograft model based on the patient organoid, and then we test in an animal. And at the end, we find the best combination of the drug, and then we can treat the patient with that drug. So I think we may be able to use microbot or nanorobot to more smart or more delicate test for precision medicine. So one of the idea for us is how to bring the microbot for body on a chip system. We are not there yet, but this is one of the general direction in our group. So we want to mimic the human by bring organoid from liver, brain, skin, or whatever we need, and also with maybe a tumor. And usually, as you know, the body on a chip system is a passive system, so we can only control the media by different pressure or motors. But if we can control the flow using microbot or nanorobot or micromotors, we may be able to make this more complicated, sophisticated, or smart body on a chip system. So one of the direction we are trying to integrate these microbot or micromotors in the body on chip system so that we can mimic in a better way for human biology. So I think magnetic manipulation, we already have several discussions. I think I can skip. So I just want to introduce. These are the system from Brad. So Minimag and Optimag, and we also developed one of the system called Maroda. Maroda is in Korean just micro robot, the first syllables of saying this is microbot in Korea. So this is Maroda. And Brad already explained that by controlling field and gradient, we can control the orientation and position of the magnetic agent. This is our system. We used integrated imaging system, so we can autonomously control those microbots. And we can control them in 3D space just because we can have two images from top and side. When we apply this technology in a larger scale for like a swine model, we can also use X-rays. As I said, my background is MEMS, so many people who I met in a conference saying that, Dr. Cho, your background is not robotics. Why you are working on robotics, or why you call it robotics? So then I tell them, you don't need to call it micro, you can call it drug delivery system or cell delivery system. So whatever is okay. So. But I believe people call it micro because we can have autonomous control to the target area as we want. So on the left side, it's untethered microbot, so which I'm going to talk today mostly. And on the right side, it's a tethered microbot, very similar to a breast catheter, but we are working on guide wire micro for coronary artery. So it probably can also apply for the neurosurgery, but right now we have a human scale control system with the 2 X-rays, and we tested with the swine models. And mostly we are developing different types of guide wire because depends on the region or depends on the property. We may need different types of guide wire, depends on the tip load, different force that we apply. So we are trying to develop different types of guide wire, and with this system and X-ray, we tested with the swine, but today let me focus more on the left side, which is untethered microbot. So I think I don't even need to mention all the previous studies because all the speakers today, they already mentioned. So basically, people working on micro or nanorobot to deliver either drug, cell, or take some samples out. So we are also doing the same thing. So in 2013, I published a paper in Advanced Materials by proposing this kind of a 3D structures, cage scaffold type microbot to deliver multiple cells or multiple drugs. So like last night in the reception, we had discussion about the research, and I mentioned that we published paper, and after we published paper, we learned one mistake. So in 2013, I thought if we can make this kind of a scaffold microbot and load cells, we may be able to deliver multiple cells. So many people are focused on single cell delivery at that time, but I thought if we actually want to use micro for medical application, we need to deliver multiple cells. So that's the idea. So we made this kind of scaffold type microbot for 3D cell culture. So this can be used for multiple cell transporter. And because my background is MEMS, at that time I was very fascinated by making this kind of structure. So the diameter of the structure was about 80 micrometer, and the length is about 270 micrometer. So it's just the size of your hair in 270 micrometer. And the importance of this research was that we were able to optimize the kind of structures inside the scaffold. Means depending on target the drug or cells, we can optimize the space to load those cells. And at that time, I thought I may be able to use this microbot for blood vessel. I mean, honestly. But later, after I published paper, and later on, I figured out that it's not possible just because this material is not biodegradable. And we were using nickel to provide the magnetic properties on the structures. And we coat with the titanium at the very left surface just to enhance the biocompatibility. So the idea was okay, and we were able to publish papers, but the biggest issue is it cannot be inserted into the blood vessel, it may cause more problem by blocking the blood vessels. So, I mean, I published papers, but I also learned something new at that time. So, anyway, with this robot, we were able to control using magnetic field. So, even in a small scale, we can have targeted control after programming the magnetic control system. So at that time we cultured hex cell, HEK293 cell, which is like more like any. I think the easiest cell to culture. So it was biologically it was pretty easy. So we published paper, but again, as I said, it cannot be used for in vivo application. So later I thought maybe it will take some time to bring this micro or nano robot into in vivo environment. So I thought, as I introduced at the beginning, maybe we can use this kind of robot for body on a chip for maybe easier practical application. So we designed the helical microbot or pole-type microbot. The pole-type microbot, the diameter is about 80 micrometer. And the helical microbot, the diameter is about 80 micrometer and length is about 270 micrometer. And we can control those microbots in microfluidic systems. So we tested. So the velocity for helical is about 500 micrometer per second. For ball type, it's about 1400 micrometer per second. And as usual, like other leading professors who deliver talk today, we can control those microbots as we want. And compared to the scaffold type microbot, which I published in 2013, the efficiency was much higher with the ball type and helical structure. So for this research, we want to have a more biological, meaningful experiment. So we used the hippocampus neuronal stem cell from mouse. So we used primary neuronal cells, and we cultured the neuronal cells on our microbot. And then we selectively differentiate those cells on the microbot into astrocyte, oligodendrocyte, and neurons. Those are the major cells composing the brain. So the important part here is we cultured the stem cells on microbot and selectively differentiated as we want. And we made a body on a chip system. So it has 6 wells. First one is to be used as an inlet. And then 4 chambers, we have liver cells, liver organoid. And then the 6 chambers, it has a cancer tissue. And the one thing that we demonstrate is we load another cancer cell on the microbot and bring the microbot from here to here at chamber 6. We bypass all other chambers. So basically, we don't need to affect those 4 other liver tissues, but we can deliver the cancer tissue into the chamber 6. So we actually did. So this is one of part of the video. We're delivering microbot from here to here. So it's a small device, but it's a long journey for the microbot. And actually, we submitted a paper after this, and it was rejected. So the reviewer said they want to see some in vivo applications. So I mean, the purpose of the research for this case was in vitro application, but we were requested to do in vivo application. So we have different approaches. So we took the red brain out and use clarity to make it transparency, because we should be able to see the robot to control in real time. So this is a red brain after clarity. So we can see the microbot in the blood vessel. So we loaded those microbot in a red brain blood vessel, and we can control them as we want, as you can see here. And another ex vivo experiment was we used the mouse brain and made a brain slice, and then put the microbot into the ventricle. So basically, assuming that if we put the microbot somehow in the ventricle, then we can control in the brain. But I mean, it maybe doesn't make sense, but at that time we were just doing our best to show the potential of the microbot. So basically, the microbot has cells and then played on a brain slice, and they can they can load the cells and they culture. But the problem is the microbeads also stay there with the cells. And the last was the actual in vivo application. So we we made this kind of sprout. So the one microbot has about 100 cells, and we used 100 microbot, and then we loaded into some underneath skin. So we were able to control these 100 microbot using magnetic field. This is the last part of this paper. But we also learned that this microbot cannot be again used for the in vivo application, because the biggest issue at that time was how to see the microbot in real time. We can control the microbot if we can see, but until now we don't have a system to use both imaging and magnetic control at the time. So we control the microbot in one hand, and then we bring this robot into imaging system to see the fluorescence images. So honestly, it was not at the same time, but I think for the real application, probably we need to have a control unit integrated with the imaging unit. So that's one of the direction that we are working on right now. So after that, I still continue that maybe how to use the microbot for in vitro applications. So next story is using the microbe for neural connections. So I mean, in general, the actual 3D in vivo brain brain connection is very difficult to study and difficult to understand. So to mimic these neural connections, people using in vitro system, like usually they predefine the pattern and then culture the cells on the predefined pathway. So the question that we had was, there's still a little known about how morphological structures in the brain interplace with the functional features. So basically, we want to control the morphology of the neural connection, and also we want to see the functional activity of the neurons at the same time. So in the previous studies, people make this kind of filler or pitch to culture the neuronal cells. And as you can see, the neurons, they can be cultured like on these pillars or pitch environment. That's because these structures mimic the ECM. However, once they have a pattern, they cannot be controlled or they cannot be changed during the experiment. And also another approach, they make this kind of group structures so they can control the alignment of neurons while they are differentiating. So later we adopted this technique on our microbot. So there are also several trials to control the neural connection in real time. So the people use AFM or patch clamp or kind of rotational structures. I think this video cannot be played. So anyway, this is rotational structures. So after they load the neuronal cell here, they rotate the kind of propeller. So they connect this structure and this structure, so they can make they can control in a dynamic way. And to read the signals from neurons, usually people use a patch clamp, mostly in biologists. And there are also some other approach from MEMS communities. They use a microelectrode array to read the neuronal signal. But this patch clamp usually has the precise readings from the neurons, but they kind of damage the neurons in real time. And this kind of a standard MIA, they can read the signal without damaging the cell, but the resolution is not that high. So what we are using is a CMOS-based MIA. This device actually developed by Professor Andrea Hellerman and ATH. So it has high-density microelectrode array using CMOS technology. So the only disadvantage with this system is it's not transparency. So we need to see from the top, not from the bottom. So we cannot use a overhead microscope. But this CMOS MIA has a very high spatial and temporal recording resolution. So both time and in the space, we can read in a single cell level in real time. This is just, I think the video is also not playing, sorry. So this is just the kind of neural recording using the high density MIA to see how accurately they can measure the action potential in real time. Is the computer frozen? It's not moving. Can you move to the next page from there? Maybe the video is not, it's still okay, but just computer has been frozen. So anyway, the idea for this research is we want to combine the microbot technology with the high density electrode array, because with the microbot we can deliver, let me try it. With the microbot we can deliver neuronal cells and we can differentiate those cells in real time. I think this should work. Yeah. Yeah, so this is the video using the high density microelectrode array. So you can see it can read the signals from the brain slice. It can read the signal from the prime neurons, and also it can read the signal from for single cell level. So we want to adopt this technology for microbot. So the idea is using microbot we can have a precise and targeted control of the cells, and we can deliver multiple cells at a time. But we were not able to evaluate the cell activities in real time. So by integrating the high-density electrode array, we were able to use microbot for active neural network in real time. Also, we can read the signal in real time. So this is our approach. So we made a microbot with a certain pattern of the group, so we can control the neural alignment. And then we predefine the neuronal cells as like here, and then we bring microbot in between the two neuronal groups. Then we culture them for about 17 days. They can make a neural network. And then everything was done on the MIA, so we can read the signal in real time. And then everything was also done in a magnetic control system, so we can control the microbot in real time. So this is, I think this is the same. I think just a two-photon laser lithography to make this kind of pattern. And then this is our experimental environment. So we use a PDMS stencil to stamp the lamine to define the predefined neuronal cells. So we culture neurons here, and then we bring microbot in between two groups, and then culture. The experiment is done on the control system on the MIA, and then everything was done in the incubator, so we can continuously control the microbot while we are culturing the cells. So this is neuronal cells on the regular glass slide, so it doesn't have any reactivity. This is after differentiation on our microbot, the neurite has reactivity. And this is real-time video, so we can bring the microbot. This microbot has cells, by the way, so we can align the position and orientation. And after 17 days, you can see that they have a nice, beautiful connections, neural connections. So this is another images. And this is after we deliver microbot in a certain area. This is reading from the high-density electrode. So we can see that those cells, neuronal cells, they are activating. We can measure the signals. So here, this is one of the examples. So our robot is here. So basically, we have a neural connection through these two groups. And yellow star means we stimulate with the electrical signal from our MIA single electrode. So in this case, we stimulate here. The signals will stay here because it has not it doesn't have any connection. And in this case, we stimulate here, and the neural signal goes through this way and pass to the other group. So as you can see, the signals goes to the other side as we activate. So I have about 20 minutes. So this is the last part of my talk today, and I think probably you saw this kind of news several times. And this is this year, August, a boy was died after swimming in a swimming pool. That's because the amoeba goes through the brain through the olfactory pathway. So that's a sad story, but we know that through the nose or through the nasal pathway, we can deliver something from the outside to the brain. So this is the idea that using intranasal delivery for microbot. So in this case, we were trying to use microbe for in vivo. So instead of making microstructures, we try to integrate stem cells with magnetic nanoparticles. So we basically made a magnetic cell microbot, and then we used the olfactory pathway from nose to the brain. By this way, we can kind of bypass the BBB, so maybe the delivery efficiency may be higher. And for human, the nasal delivery is already being used in hospital for treatment. So we made stem cells integrated with nanoparticles. So this is a fabricated cell ball. By the way, this is human-derived human-derived stem cells. And as we have the nanoparticles, we tested the differentiation, and we can confirm that the cells with nanoparticles still has all the functionality for the original cells. They can differentiate whatever they can. And then we made a spheroid, so we can control them in a microfluidic environment. And this is a cortical brain organoid. This is our cell bot, and they can be integrated and grow without any problem. So in this case, for the control group, we used the same cell bot, but we didn't apply external magnetic field. For experimental group, we used the same cell bot, which has magnetic nanoparticles, and we applied the magnetic field for 2 days. So after 2 days, you can see that the control group without any magnetic field, they lost all the cell bot, which has magnetic particles. But with the experimental group, they still have these cells. So this is more like a qualitative study, not quantitative study. So if you ask me how many of cells delivered to the brain, I don't know yet, but we are still undergoing. But it's for sure we can say that with the magnetic field, we may able to increase the retention rate of cells, I mean magnetized cells in the brain. So this is one of the directions that we are working on now. So compared to control, in an experimental group, we can see the fluorescence stem cells with the magnetic particles after experiment. So I think that's done. So the time is over, so I've skipped all the conclusion. But one of the important messages that I have now is how to see the microbe. So we all discussed how to use microbe, how to develop microbe, but I think one of the most important part is how to see the microbe, especially when it goes to the nanoscale, how to see the nanorobot in real time for real in vivo applications. So this is one of the important questions that we have, and we are trying to develop some of the system for real-time imaging, without using X-ray or using ultrasound. All different types of the imaging modality have a pros and cons. But for fluorescence, of course, the penetration depth is not high. And X-ray, we can use it, but the size can be seen up to maybe 300 micrometer or above. But it has X-ray, so it's a disadvantage. PET-CT, I think, is semi-used, but just basically we don't have that system. It's too expensive. So we are trying to develop other imaging modality for real-time control of the microbot and nanorobot. And that's all done by students and researchers, and those are my collaborators. Not all, but most of the collaborators. So that's all my talk, and I'll be happy to answer if you have questions.
Thank you very much. Yeah, we can take 1 or 2 questions before we finish.
Yeah, thanks. Very nice. I'm curious about this intranasal.
Yeah.
So you might be circumventing the blood-brain barrier, but does that mean you need to go via a neuronal route? Are you going along nerve cells then? I mean, because it's clearly not an open hole, right?
Yeah, I don't know the details of the anatomy, but I was working with ENT doctors for this project, and they already regularly use intranasal pathway to deliver cells to the brain. But the
cells?
Yeah. So here we use the cells, integrated cells with the magnetic particles.
But do you hit any barrier, or do you go along nerve canals? I mean, it's got to be one or the other. No?
I think it's along the nerve canal. So there's already a track from nose to the brain. So we use the existing track.
System. Okay. Thank you.
Thank you.
I have you just I think bring up a very important point that how do you image micro robots or nano robots in real time? I think that's a good point. And so what I mean, we have, and people are using CT in general, and perhaps PET. But what is your thoughts on that? And what would be the alternative way of real-time imaging?
We don't have much progress yet, so I cannot tell the detail. But basically, we are trying to use short-wave infrared. Short-wave infrared, the penetration depth is limited, of course. It cannot be used for heart or maybe deep brain. But with good sensor and high power laser, we may be able to see up to a couple of centimeters or maybe maximum 10 centimeters. In that case, I briefly introduced our guide-wire microbots. But in that case, maybe we can use those guide-wire micro at least for the legs or arms without any X-ray.
Okay.
And also for at least for the animal experiment, like a rat and mouse.
Okay.
That's 5 centimeters, maybe good
all
enough.
Right. Yeah.
So that's one of the approach.
All right. I got it.
Okay.
Thank you.
So there is one very last question. Okay. Go ahead.
Hi, this is Ju-Young Kim from one of the students of IBS CNM. I have just a quick question. You showed us a great work about delivering micro robots and recording the neural networks. And I just have a question about if there's any interference or noise between the recording the cell signals and the magnetic field of electromagnetic system at the same time. Yeah.
So the interference between the magnetic field and reading the electric signal from the cell.
Yes, yes.
So that's, I think, important question. But because of that, we didn't do it at the same time. We deliver robot, and then we can turn off the magnetic field, and then we can record the action potential.
Okay. What if we do at the same time? Would there be any noise or interference?
We confirm that the magnetic field, it doesn't affect to the cells, and we also affect it when without cells when we apply the magnetic field. We didn't see any noise, but we didn't try it with the cells. So that's probably, I cannot answer at this moment, but probably that's one thing that we can try.
Thank you.
Okay, thank you very much. Let's thanks again Professor Che for his wonderful talk.
Thank you very much.
Then this is the end of the session, and there will be announcements.
Hello, this is the final time for the announcement for today. So thank you very much for your participation, everyone. And after the session, you have a couple of less than about an hour to have a break. And then for the invited speakers only, there'll be a dinner arranged for you. So we will meet you at 5:30 at the lobby, please. So please come to the lobby by 5:30. And tomorrow, for the invited speakers, I will meet you at the lobby again by 10 to 8. Okay? I will meet you at 10 to 8 at the lobby. And we are going to take a bus and go to Yonsei University for the second day. Thank you very much indeed.